Good morning for everyone. I'm very happy that we may meet at the Institute of Dendrology of the Polish Academy of Sciences. Please let me introduce Godziński, and I am the director of the Institute. And we are very happy that you came to our institute, to our arboretum, and to join with us this special moment for us. It is my great pleasure and honor to welcome all of you at the second International Conference for Young Scientists, Research and Practice in Forest Ecology. We are very happy to host you at the Institute of Dendrology of the Polish Academy of Sciences and to bring together such a diverse community of young scientists. The main goal of our conference is to create a space for dialogue between researchers and practitioners or research and practice. Forest ecology is a field which scientific knowledge has direct importance for the management, for nature conservation, nature protection, and restoration of forest ecosystems. Today, when forests face growing pressures related to climate change, to biodiversity loss, invasive species changing land use, and increasing social expectations, cooperation between researchers and practitioners is more important than ever. This conference is intended to support the exchange of knowledge, experience, and ideas. We hope it will be inspired for new collaborations, help identify practical solutions, and strengthen the links between ecological research and the real-world forest management. At the beginning of our conference, I would like to sincerely thank to the patrons of this conference. I mean, Minister of Science and Higher Education, the President of the Polish Academy of Sciences, the Director General of the State Forests, General Directorate for Environmental Protection, and the Mayor of the town and municipality in Kórnik. And at the beginning of the speech, I would like to kindly welcome our guests. I would like to kindly welcome Dr. Dariusz Grzybek, the President of the Zakłady Kórnickie Foundation. I would also welcome Mrs. Violetta Nawrocka, the Director of the Wolin National Park. At the same time, I would like to welcome Mr. Wiesław Krzewina, the Director of the Regional Directorate of State Forests in Poznań. The mayor of the municipality and town of Kórnik was not able to come with us, but the deputy mayor is with us, and I would like to kindly welcome all of us also. I would also kindly welcome the head of the Kórniczanin journal. This is the local journal from our city. I mean, Łukasz Grzegorowski. And we are also very grateful for the support from the Radio Poznań, as you can see on this picture. Thank you so much for coming and being with us. I would like to say a few words about my thinking of the work we have conducted for the last couple of months. I would like to thank all of you, the mentioned people, for your support, for the trust, and for recognition of the importance of this event, the second conference at the Institute of Dendrology of the Polish Academy of Sciences. Your patronage gives special value to our meeting and confirms that significance of forest ecology for science, environmental protection, and society is really very important. I would also like to warmly welcome our keynote speakers from the Institute. I am grateful that you have accepted our invitation to present your studies and that you will share your knowledge, experience, and perspectives with us. A very warm welcome also gives to all participants. Thank you for being here, for your interest, and for your readiness to contribute to discussions. Each of you brings a valuable point of view, and we believe that the conversations held during this conference will be as important as the former presentations. The conference participants are from 16 countries, namely Algeria, Belgium, Brazil, China, Croatia, Czech Republic, Estonia, France, Greece, Georgia, Germany, Poland, Serbia, Sweden, United Kingdom, and Italy. 16 countries in total. At the same time, I would like to kindly thank to the scientific committee and organizing committee of our conference. Our people, our staff from the Institute has spent a lot of time to prepare everything, and I'm really very grateful to all of you. Thank you so much. During the conference, we will take part in plenary lectures, oral presentation of the participants, and discussions devoted to various aspects of forest ecology. The program will address both fundamental research and practical applications, including forest biodiversity, ecosystem functioning, climate change, adaptation, conservation, restoration, and sustainable forest management. You will also visit Wolin National Park, about the field excursion. You will also visit the Kórnik Arboretum, also the field excursion. Our labs, you will take a part in workshops organized by the staff, research staff from the Institute of Dendrology on Friday, if I remember well. We also hope that the breaks, informal meetings, and accompanying events will provide good opportunities for networking and the exchange of your ideas. I hope that this conference will be not only scientifically fruitful, but also enjoyable and inspiring for you and for us. I wish you all interesting presentations, discussions, new contacts, and a pleasant stay at the Institute, in Kórnik, and in Wolin National Park. With these words, I am honored to officially open the second conference, International Conference for Young Scientists, Research and Practice in Forest Ecology. Thank you so much, and you are very welcome. Thank you. So we have about 20 minutes to present some information about our institute and about the Kórnik Arboretum, which is the oldest part of the institute. Because you will have some time to spend at the Kórnik Arboretum, thus I have prepared only a few information about this place, which is really very important, not only for the Institute's scientists, but also for people from Poland and other countries. And I hope you will agree with me after visiting the Kórnik Arboretum. Let me describe the Institute and our oldest part. As I have mentioned before, Kórnik Arboretum is one of the most important parts of the Institute of Dendrology of the Polish Academy of Sciences, and it is the oldest part of the Institute. It is not only a historic garden, but also a living collection of woody plant species, trees and shrubs, used for research, for education, for conservation. In this short lecture, I would like to show you why the Arboretum is a real heart, as I have mentioned here, for our institute, and not only for our institute. Although this year is very important for us because we are celebrating the 200th anniversary of the Kórnik Arboretum, the history of gardens in Kórnik is much longer than 200 years. Since the Kórnik Castle, you will see the Kórnik Castle in the evening, itself is celebrating 600th anniversary this year, we can be sure that the gardens around the Kórnik Castle here is much older than we expect. So in fact, it is more than 200 years. However, we will not go so far. This year is a very special moment for the Institute and for the Kórnik heritage. We are celebrating several important anniversaries. I mean, the 16th anniversary of the Kórnik Castle, the 200th anniversary of the Kórnik Library, the 200th anniversary of the Kórnik Nurseries, and the 200th anniversary of Kórnik Arboretum of the Institute of Dendrology of the Polish Academy of Sciences. The 19th century owners of the Kórnik estate showed great interest in dendrology and in natural sciences, and it was thanks to them, I mean about these two owners of the Kórnik Castle, thanks to them that a very rich collection of woody plant species was gathered in Kórnik Castle Garden. Although the beginnings of Kórnik Gardens go back much earlier, the true founder of the Kórnik collection of the Arboretum was Count Tytus Działyński on the left. Titus was a very important figure for Wielkopolska region and not only for Wielkopolska. He was a collector, a bibliophile, publisher, and above all, the founder of the Kórnik library and the Arboretum. When he took over the Kórnik estate in 1826, he not only rebuilt the castle, but also completely transformed the surrounding gardens around the Kórnik castle. The first confirmed purchases of plants for the Kórnik garden date back to 1826. Titus initiated the creation of the woody plant species collection, and Jan, his son, greatly extending the work started by his father. Jan Działyński, the son of the Titus Działyński, the Count Titus Działyński, amassed a collection in Kórnik that included around 1,500 species and varieties of trees and shrubs not far from the Kórnik castle. Jan Działyński died without children, and he passed the Kórnik estate on his nephew, Count Władysław Zamoyski, the last owner of the Kórnik castle, of the Kórnik estate and Zakopane estate. His mother in the middle and his sister on the right successfully implemented and developed the intentions of his uncle and grandfather. According to the last will of Count Władysław Zamoyski, in 1925, the Zakłady Górnickie Foundation was established and the Zamoyski's estate was handed over to the Polish state, to the Polish nation. It was really very important for us, and it is still important for us. Zamoyski's will was to establish and maintain a research center in Kórnik, both on the mountain slopes and on the plains, for the study of everything that falls within the scope of breeding, living, protection, and proper exploitation of all kinds of trees, both domestic in the country and foreign ones, that may be usable for country, forest, garden, usable fruit and ornamental trees. Their wood, fruits, leaves, juices. Care to the Kórnik gardens. The first director of the Kórnik gardens since 1926 was Antoni Wróblewski on the right. He was an outstanding dendrologist and researcher. The collections were organized and greatly expanded in his days. The director of the Kórnik Gardens established wide cooperation with botanical gardens from all over the world and nurseries in Poland and abroad. Many new plants arrived in Kórnik, and the Kórnik nurseries developed very dynamically. New cultivars were selected, orchards were expanded, and detailed documentation of the park was created in those times. Thanks to Antoni Wróblewski, the first director of the center of the Kórnik Gardens, the collection included almost 4,000 tree species by 1936. At some perturbances, on July 1st, 1933, a scientific institution was established in Kórnik. It was the Center for Research of Trees and Forests, although the establishment of only one of the 4 plant divisions was successful on the left. The first director of the Center for Research for Trees and Forests was Antoni Wróblewski, in fact, who had managed the Kórnik Gardens of the Zakłady Górnickie Foundation since 1926. Rapid development of the Institute of Dendrology, today's Institute of Dendrology of the Polish Academy of Sciences, took place in 1952 when our institution was incorporated into the structure of the newly created Polish Academy of Sciences. It was 1952. In 1974, in recognition of the level of research we conducted at the Institute, at the department, in those times, our institution was elevated to the rank of the Institute and is called right now Institute of Dendrology of the Polish Academy of Sciences. We use this name to this date. The mission of our institute was the same exactly for more than 90 years. To carry out scientific activities in the biological sciences and forestry, in particular those relevant to national development, and to disseminate and implement the results of these activities. Today, we conduct research in teams that include 5 scientific departments. They are Department of Biogeography and Systematics, Department of Developmental Biology, Department of Ecology, Department of Genetics and Environmental Interactions, and Department of Symbiotic Associations. Of course, we have more departments at the Institute. I mean about the Arboretum and Experimental Forest, Library, Laboratory of Mineral Analysis, and the last but not the least, the Herbarium. The collections gathered in the Arboretum were and are extremely important, not only for the Institute staff, Institute people, but this is an important object for our research in Poland and in other countries, because we have a wide cooperation with many institutions abroad and in Poland. The Arboretum is the Institute's flagship. The Arboretum's collection of woody plants is one of the largest and oldest in Europe, in this part of Europe, and is constantly being expanded by the Institute. It is, however, not only a place of research, but also a thriving center for tourists and knowledge promotion activities. I didn't mention yet, but the Arboretum is a botanical garden. So that is a specially arranged and managed area according to Polish law, together with its technical infrastructures and buildings. It is a place for ex situ conservation, cultivation of plants from different climatic zones and habitats, cultivation of selected plant species, as well as scientific research and education. These are the really important goals of the botanical gardens. The main tasks of the botanical gardens include taking part in scientific research aimed at protecting species threatened with extinction in the wild, education in the field of plant, animal, and fungi conservation, including the protection of biodiversity. The third one is cultivating endangered plant species for ex situ conservation, and when possible, of course, reintroducing them into nature as part of the conservation programs. Keeping plants in conditions suitable for their biological needs, and the last but not the least, the maintaining proper cultivation records. In Poland, there are more than 40 institutions like that, 40 botanical gardens distributed across the whole country. We are one of them. Today, the botanical gardens are a global network of the institutions. Their number has increased from about 2,500 botanical gardens to 1,000 more. They employ thousands of botanists, the key specialists in plants functioning, and maintain very large living collections, including tens of thousands of plant taxa in the world. Their main roles are scientific research, plant conservation, citizen science, and sharing knowledge with society. And we do all of this task also at the Institute in our Arboretum. 2/3 of botanical gardens worldwide are located outside of the world's 36 biodiversity hotspot, the most important parts in the world where the biodiversity is really created and worth to conserve, to protect. The botanical gardens are important places for scientific activity and for scientific research. Interest in them is steadily growing, as you can see on this picture. Recently, about 2,500 scientific articles are published every single year based on research carried out in or with the material from botanical gardens. In recent years, research conducted in botanical gardens has become especially important in relation to climate change. This research focuses on understanding the effects of climate change on living organisms, not only on plants. So this is really important, not only as a scientific institutions, but also, as we have in Poland, a lot of botanical gardens in the structure of state forests that are used for the same purposes. At this point, I would like to mention that the last year, in 2025, our Arboretum was ranked 7th in the world and 2nd in Europe in the ranking of the top most referenced arboreta in research worldwide. This is a source of pride and satisfaction for us, of course. And a few words about the area. Today, the Arboretum includes about 33 hectares of the old castle park and about 16 hectares of the so-called new Arboretum not far from the main buildings of the Institute. However, the name new may be misleading because collections have been established there for 80 years. One of the missions of the Arboretum is to collect woody plants. The Arboretum functions as a botanical garden, and I have mentioned before, and belongs to the network of Polish botanical gardens and arboreta. At the same time, it is a historic garden and part of the historic monument complex, together with the Kórnik castle, as you will see it during the conference, and the Kórnik church in the city center. The collection includes over 3,000 taxa of woody plant species. Each year, we add new items to our catalogs, but unfortunately, each year we also lose some of them. The Arboretum is a place where scientific research is carried out, both by employees of the Institute of Dendrology of the Polish Academy of Sciences, and also by researchers from other institutions. The Arboretum is used as a source of research material for many research works. Depending on the needs, we collect and provide materials such as shoots, such as leaf buds and seeds, and observations are also carried out not only also in the grants. The Kórnik Arboretum has many rich collections, and we will speak about them in more detail during the field excursion, in the field. Here I would like to draw attention to just one of them, the National Collection of Lilacs, which includes over 250 accessions and has the status of the national collection in Poland. The establishment of this collection began already in the interwar period, and today it is one of the largest lilac collections in Europe and certainly one of our most beautiful collections now, and you will see the collection. Our Arboretum is an excellent place for education. The collection offers very good opportunities for teaching, and our audiences are also very diverse. We run classes for students, mainly for forestry students and horticulture students, biology students, but also for the future architects. We also organize internship for university students and secondary school students. Each year, the Arboretum is visited by more than 100,000 of people. We organize thematic events, exhibitions, and guided tours in the Arboretum. The Arboretum is also a place for sports and cultural events. Educational materials are available for visitors of different ages. There are also many information boards in the Arboretum and thematic path, so you will also see all of them. So far, we have collected, studied, thought, and shared, and these activities show that we have achieved and how we have built our experience. In the future, we want to continue in the same direction. I mean, to collect, to study, to teach, and to share, developing knowledge and making it useful for others. And the question is why? Because we all love trees. At the end, I would like to kindly thank the Ministry of Science and Higher Education and the Zakłady Górnickie Foundation for financial support. Thank you very much, and please say my gratitude to all of the people from the foundation. And we are also very grateful to our patronage, as you can see on this slide, the Honorary Committee, and the media patronage for support of this event. If you would like to read more information about the Kórnik Arboretum, not only about the Institute, because about the Institute I was talked 2 years ago during the first conference, it is possible to read a short information about the Kórnik Arboretum, reading the book of abstracts. The most important things are available in this text. Thank you so much for listening. Thank you so much. And now I would like to ask Professor Ewelina Ratajczak to conduct the conference. Thank you.
Thank you, Director Jagodziński. I would like to welcome you all. Now we begin the keynote lecture session. We have 14 presentation today. The session is chaired by Professor Ewa Kalemba and Professor Marcin Pietras. Today talks will cover a wide range of topics in forest ecology, from fundamental research to modern approach and future perspectives. I hope the session will be interesting and inspiring. Thank you, and I invite Professor Ewa Kalemba.
Good morning. We will start the first session, but before the start, I have one information related to the, because we have 20 minutes for each speaker. That's why me and Professor Pietras will inform you about the time that it is passing. We will use the skills of Professor Pietras, who is also the professional football referee. That's why when it will be the 18th minute of the presentation, Marcin, can you stand up? This will be shown to you. It's the beginning. Then it will be the 19 minutes. And again, the yellow card. But for the 20 minutes, it will be the different color. Yes. So we want to keep the framework of this session. That's why I would like to invite the first speaker. The first speaker is Professor Dominik Tomaszewski, and he will talk about the importance of the integration of data from different sources like herbaria, citizen science observations, also global platforms, and the importance in life across the time and space. The title of the presentation is Big Data Fuels a Botanical Time Machine. So Dominik, the floor.
Thank you very much. Thank you very much. Good morning, everyone, and thank you for being here. Today's topic is big data and botany. So there will be plants, there will be data, there will be a few very large numbers, and I try to make sure that large numbers behave themselves somehow. And big data is changing biology, and of course modern botany as well. Not just little, but quite deeply. But actually, what do we mean by big data? It's not just many rows in Excel. Even if we seem to enjoy very large spreadsheets, big data means that the data are so large, so complex, and so diverse that we need dedicated IT tools and statistics to work with them. In the past, much of biology was based on local observations. As a researcher went into the field, studied one site, one species, or one community. This is still essential, but now we can also ask questions at much larger scale, across continents, across decades, or even centuries. This is possible because biological data now come from many different sources. The first is biological collections. For us, this mainly means herbaria. Each specimen is a real plant, and it's like a small piece of evidence from the past. We have citizen science, and modern platforms allow millions of people to record plants with their phones. This is very useful, but sometimes they also can photograph the same tree 500 times. When we have environmental data, like climate, topography, land use, satellite images, and so on, to these we can add monitoring programs, ecological studies, molecular data, trait data, or large image data sets analyzed with AI. But the real power not comes from one source alone, it comes from the integration. And thanks to it, we can see patterns that no single researcher could see alone. This is why modern botany is no longer just about plants, it's about data systems that connect the past, the present, and the future. I like to think of it as a botanical time machine. Herbaria take us to the past, citizen science shows us the present, and models help us to understand the current processes and and anticipate future changes. Let's start with herbaria. They are still one of the most important sources of data for botanists. There are about 400 million specimens in more than 4,000 scientific herbaria now. And these numbers keep growing. Each specimen is a piece of evidence, and taken together, they form a huge archive. Of course, herbaria are central to plant systematics and play a key role in discovering new species. It's been estimated that more than half of the species still to be described for the science have already been collected and housed in our herbaria. From big data perspective, temporal depth is a key strength of herbaria. Many collections go back more than 200 years, and this allows us to study aspects impossible to analyze using other sources of data. And herbaria have changed considerably over time. They've traveled a long way from medieval manuscripts with drawings through volumes with pressed plants glued onto pages called ortisicci or dry gardens to what they are now. And now, a modern herbarium is not just a set of cabinets. It's a research infrastructure with proper storage, databases, and technical support. Today, digitization should be a part of every herbarium. It allows collections to be used not only by people who can visit them in person, but the core idea of a herbarium hasn't changed. They are for documenting and studying plants in a structured and lasting way. And the good old herbarium sheet hasn't changed much either, probably because it's based on a simple idea that still works very well. A herbarium specimen follows a very clear basic format: a plant, a sheet, and a label. The specimen itself gives us direct biological data. We can see its morphology and micromorphology, which is essential for identifying and comparing species. We can extract DNA so it can be used for genetic studies. We can analyze its biochemistry, and quite often it's more than just the plant. There may be traces of herbivores, fungi, or other organisms. So in a way, we don't collect the plant, we collect a small ecosystem. And without the label, the specimen is just a pressed plant. The label tells us when and where it was collected, often quite precisely. It tells us who collected it, and sometimes more, like in what kind of habitat or what other species were present there. And of course, it provides the scientific name, which is so important for taxonomic system. When we combine these two elements, I mean the specimen and the label, we unlock a wide range of research possibilities. In systematics, morphology and DNA allow us to classify organisms. If we add location and time, we can move into population genetics and phylogeography. And with many specimens together, label data become especially powerful. We can map species distributions, build models, and study biodiversity patterns. Habitat information helps us understand where the species lives and how it interacts with others. And there's time too, because every specimen has a date. We can study phenology and detect shifts in species ranges, which is so useful when studying climate change. Let's don't forget about the conservation biology, because they help us see where species are declining or expanding. And finally, herbaria tell us a human story as well, as they are useful for history of science. And so in short, a herbarium specimen is much more than a dried plant. It's a multidimensional source, biological, geographical, historical, and taxonomical at the same time. If herbaria are about the past, then citizen science is very much about the present, and it's moving fast. What really stands out here is scale. Platforms like iNaturalist or eBird produce millions of observations. Each one usually has a location, a date, or often a picture. So instead of going out and collecting a few records, we suddenly have the data coming in all the time and from many different places. It's a bit like having thousands of people in the field just without the logistics. And that scale really changes a lot. We are no longer limited to studying one species at a time. We can model distributions for many species at once, and we can do it with much better spatial detail. Technology also plays a big role here. So, for example, automatic species identification from images. Machine learning models used in apps in our smartphones are trained on huge image data sets, and they improve as more people use them. This makes species identification faster, easier, and more accurate, of course. And because everything is time-stamped, we can also look at species phenology, like when plants flower, and how this changes over time. This becomes especially useful when we think about climate change again. So overall, citizen science makes research more dynamic, more detailed, and more collaborative. So, as I said, individual sources of data are incomplete, but together they form something much more powerful. And now we have systems that bring these data together and turn them into a research infrastructure. And right now, the most important data aggregator in this data ecosystem is the Global Biodiversity Information Facility, or GBIF simply. It was set up 25 years ago as an international initiative with a very simple idea: to make biodiversity data available and bring them together in one place. In practice, institutions publish the data in a standardized way. This means that records from different sources can be combined within one global system. As a result, GBIF has become a huge global database. Currently, it contains nearly 3.7 billion occurrence records. If we look just at plants, there are over 560 million records. More than 130 million of these come from herbarium specimens, and over 55 million already have images attached. And these numbers are still growing very quickly, largely because of ongoing efforts to digitize biological collections. But before we get too excited about these huge numbers, we should ask one simple question: how reliable are these data? And this slide shows so-called sampling bias. It represents area change relative to sampling density. In simple terms, areas shown in orange, red, and black are oversampled for the size, while areas shown in green and blue are undersampled. And the message is clear: big data is not evenly big everywhere. We have many records from places that are easy to reach, but remote tropical forests, mountains, and many regions of the global south are still much less visible. This applies to herbarium collections, but also to citizen science. Botanists collected where they could travel. Citizen scientists record where they live, work, or spend their holidays. So a map of plant records is not only a map of plant biodiversity, it's also a map of botanical activity, because they show not only where plants are, but also where botanists had good roads or just funding. This doesn't make the data bad. That means that we need to read them carefully. And always you should ask which areas are oversampled, which are missing, and how this might affect our conclusions. This slide shows temporal bias. In simple terms, herbarium collecting was not constant over time. There were quiet periods, and then there were large waves of collecting. Some years are full of specimens because there was an expedition, a taxonomic project, a new institution, or more funding. Other years are almost empty. These waves also happen at different times in different regions. So herbarium data are not just biological records. They are influenced by wars, migration, economic change, or research institutions and collecting permits. Citizen science has a similar problem, but on a shorter time scale. Most observations happen in spring and summer, when plants flower and are easier to recognize. They also increase at weekends or during holidays. So a data set doesn't show only where species really are. It shows when and where people were able to make records. And apparent increase in our records may suggest that species is spreading. But it may also mean that there were more collectors, more observers, better apps, or simply better weather. That's why we should interpret trends in big data carefully. On this slide, I'd like to show you an example of collector bias. This pattern is very simple. Most collectors collected only a small number of specimens, and a few people collected enormous numbers. These are the mega collectors. So we can say that the data set was shaped very strongly by a very small group of extremely active botanists. And this matters because every collector has habits. They may visit particular places or prefer particular plant groups. Some people focus on trees, some on orchids, and some on difficult taxonomic groups. If one person collected tens of thousands of specimens, the habits become part of the dataset. Citizen science has a similar problem, which we can call trait bias. And big plants, beautiful plants, easy to recognize or rare plants, they all attracted attention. Small, boring, or difficult plants are less lucky. But bias is not the end of the story, it's a part of the story. And we understand, if we can understand how the data were collected, we can use large data sets in much more careful and effective way. As I said, big data also shows us the history of botanical collecting. At this point, I would like to tell you about our study, in which we used GBIF data and looked at the global distribution of holotypes. When a new species is described, one herbarium specimen has to be chosen as a reference for all future identifications, and it defines a species name. That's why holotypes are among the most valuable objects in our herbaria. So we linked the place where holotypes are stored today with the place where they were collected. We analyzed about 120,000 GBIF records from digitized herbarium collections. The first thing we can saw was that most holotypes come from regions of high biodiversity, especially the tropics. In fact, 76% were collected within 30 degrees from north and south of the equator. This fits very well-known pattern, the so-called the latitudinal gradient of biodiversity. In other words, species richness decreases as we move away from the equator. But at the same time, the great majority of holotypes are now stored outside that zone. This suggests that they were moved from the site of collection to other parts of the world. Here on the left, we have the regions where the specimens were collected, and on the right, we have the regions where they are kept now. And one thing is very clear here. Material from North America, Europe, Australia, or New Zealand mostly stays within these regions. But for other regions, the pattern is quite different. In Africa and Asia, more than 90% of holotypes are stored outside the regions of origin. In Central and South America, it's a bit lower, but still high. It's around 77%. We looked also at how this changed over time. For each year, we calculated the proportion of holotypes retained within the region of origin relative to the total number collected in that year. Thank you. What we found is quite striking. The most dynamic changes occurred in regions with high biodiversity, namely Africa, Asia, and Central and South America. In this region, the share of holotypes kept locally increases over time and quite rapidly. The pattern is clear, and the increase is close to exponential. Australia and New Zealand behave slightly different. For a long time, for the early material was sent abroad, mainly to Great Britain. But in recent decades, we have seen a rapid shift, and almost all the holotypes are now staying in the region. So overall, these results show a clear shift. In the past, many herbarium specimens were moved to Europe and North America. This was partly a result of colonial history. Today, more and more holotypes are kept in the regions where they were collected. We think that this reflects the growth of local scientific capacity. If there are more universities, more specialists, and more scientific herbaria in the regions, more new species can be described there, and the holotypes can stay close to the place where the plants were collected. This is just one example, but it shows the broader point. When we bring many records together, we can see not only where the plants were collected, but also how science itself has changed over time. And that brings me to the final message. Big data in botany doesn't make our data sets bigger. It changes how we think and how we work. We move from simply describing patterns to predicting them, from local observations to global models. And of course, this comes with challenges. Our data may be biased or messy, but despite that, something important happens. When we combine herbarium records, field data, and citizen science, we can start to see a bigger picture. And this is really the key message. We can turn scattered plant records into a kind of botanical time machine. And it's a very powerful tool, but it's not automatic. It runs on slightly imperfect data, and it still needs calibration. Big data doesn't replace biological thinking. It only works when large data sets are combined with real knowledge of botany. And then it gives us a better way to understand where plants came from, where they are now, and where they may be going. So now we have more data, better tools, and bigger questions, and still somehow plant names remain difficult. Thank you very much.
Thank you very much for the very interesting lecture. According to the conference program, right now we were supposed to have the lecture by Katarzyna Sękiewicz with the title Genetic Diversity and Hybridization for Forest Resilience. However, she couldn't join us today. That's why we will skip this lecture and move on to the next point, to the next lecture. That's why I would like to invite Professor Emilia Perskamczyc. She will talk about the complexity of plant reproduction, in particular the reproduction of trees. And she will focus on dioecy, which is complex but interesting. Will be, does plant sex matter? And the continuation of the title, Dioecious Plants in the Face of Environmental Change. You can begin.
Hello, everyone. As it was mentioned, I would like to tell you something about plant sex. It's matter or not. We all know that, well, I assume that we all know that plants have different types of sexual systems. Most of them are hermaphroditism. It's the case where both male and female reproductive organs are present in the same flower. There is also monoecy, where plants have separate male and female flowers on the same individual. There are different also systems like gynodioecy, where both hermaphroditic and female-only individuals are present in the population. Today, I would like to focus your attention on dioecy. It means that we will focus on the species that are characterized by separate male and separate female plants in the same species. So this sexual system in plants, it's present only in 6% of all world's flora. However, it's really important that in gymnosperm, 65% of all species are dioecious. According to classical sex ratio, we should have 1-to-1 female and male plants in the population if the cost of reproductive and growth are similar for both sex. However, we observe that we have deviation. We don't have one-to-one in natural population, and this was connected with environmental factors. Drought, so declining total precipitation, warming temperature, and fragmentation increase one or favor one of the sex. So in drought condition, we will have much more male plants compared to the female in the population. And when the environment, it will be rich, and we will have much more female plants. According to literature and our observation, those species are characterized by the presence of sexual dimorphism, and it was written that females are characterized by lower growth rate, greater mortality because of this stressful condition in the environment. They have different storage strategy, and it was pointed in many of the articles in the research that females are described as the most sensitive individual within the species. We all know that we have different threat for plants. We have drought. We observed lower precipitation in different areas of the world. However, recently also nitrogen deposition is pointed as one of the threat for the plants. Although it was described and in different environments is described as primary limiting element that affect the plant growth. However, in different areas of the world, there is higher deposition of this element observed, and it was related to decrease in plant species richness, higher plant growth. However, we observe also different impact on seed quality. And according to different models, we will have much more nitrogen in the environment, and it will decrease even that we decrease in nitrogen use for the fertilizers, as fertilizers. According to resource allocation hypothesis, plants will manage with all of available resources to vegetative growth, defense, or reproduction. And if we have great investment in one of those paths, we will have less resources to invest in other direction. So for me and for many of our researchers from the Institute, we were wondering how availability of the nitrogen will impact sexual dimorphism in dioecious plants. Many of I put the description of many of the articles published by our researcher in the book of abstract. Today I will focus just only on the one topic, so I would like to present data more detail related to effect of nitrogen deposition on the reproductive potential of woody plants. So we wanted to check how nitrogen fertilizer availability will impact on seeds quantity and quality, seedlings quality, and pollen grains production and quality. We use different features to characterize those, to answer the questions, but we selected two model plants. So we use Juniper communis and Taxus baccata in species. So they are both dioecious plants. They are characterized by secondary sexual dimorphism. They are wind-pollinated plants, evergreen and gymnosperm. So they are really similar. However, Taxus baccata produce seeds within the 1 year and prefer rich environments. Juniper produce seeds within 2-3 years, depends on the environment, and it prefers and it's present in poor environmental condition. We can describe those species, those individual sex by checking the presence of alleles or berries or flowers, male or female flowers on the shrubs. So for this, we perform experiment in which we took shoots, we rooted them, and we produce clone plants from both of the species. And we started this in 2012, and in 2013 we randomly selected plants. So remember, keep in mind that it was male and female individual plants, the same genetical diversity because they were clone plants, and they were kept in the same condition of everything but not fertilizer addition. So half of them, half of female and male plants, received fertilizer addition once per year with the amount of 5 grams of fertilizer called osmocote, so it was 50% of nitrogen. And the rest part of the plants didn't receive any fertilizer. And we kept those plants from 2013 up to 2018. We observed them, we analyzed them, and then we were happy because they produce flowers and they brought and produce seeds. So then we could analyze how fertilizer availability will impact on plant growth. We observed that fertilized plants, of course, were grow higher. They were they produce longer main shoots. They have similar number of branches, but longer lateral branches, and they produce seeds. And the production was depend on the height of the main shoot of plant. However, this relationship was only revealed in the non-fertilized condition. When we checked how those plants produce seeds and how many of them are mature or ready to do it, we observed that fertilized female plants produce seeds earlier than non-fertilized plants. And this, the difference was around 1 month, and it was present in both of species. When we wanted to check how the fertilizer availability will affect the seeds quality, so we checked the first easy to measure mass of the seeds. So we observed that, of course, fertilized plants produce much more seeds. The seeds mass was heavier, however, we also observed that seeds were efficiently produced by non-fertilized plants. On the beginning, we counted the numbers of flowers produced on each shoot on the female plants, and after we observed and collect and count seeds, number of seeds. So we observed that non-fertilized plants produce seeds with much higher efficiency than non-fertilized plants. We also checked the content, so the quality of the seeds. We observed that there were differences in carbon, nitrogen, and carbon to nitrogen ratio in those seeds, especially when we look on the Juniperus communis characterization of those features. We also wanted to ask if we observe the nitrogen amount increase in the seeds, if it's present also in the metabolome and how it's collected and stored in the seeds. So we analyzed the metabolome of seeds collected from those two species from both of the groups, and we observed that 80 metabolites were changed in Juniperus communis seeds. However, none of them were changed in Taxus baccata in relation to fertilizer availability. So Juniper communis was much more affected by fertilizer availability compared to Taxus baccata. After that, we also wanted to check germination ability of those seeds. So we used only Taxus baccata seeds. Thus, Juniper communis was not so numerous, let's say. So we germinate, stratify them and check the germination rate, and we observe that fertilized plants produce seeds that were characterized by lower germination rate compared to those non-fertilized compartments. And the seed size also affect the germination ability. Non-fertilized plants produce much more medium-sized seeds, and they were better in those parameters like germination ability. We also check the 3 months seedlings to describe how seed size and seed origin will impact this growth. And we observed that much more impact was placed in relation to seed size than compared to fertilized or not fertilized condition. So we observed that this condition will impact reproductive potential of female plants. Then we wanted to also check how nitrogen availability for long term will impact reproductive potential of male plants. So we wait until they produce flowers, produce pollen grains, and we collect pollen grains and start to germinate it in vitro condition. So we observed that Juniperus communis produce pollen grains that only 3% of them start to germinate in vitro condition compared to 37% of non-fertilized pollen grains. And similar but not so huge difference were observed also for Taxus baccata, pollen grains produced by those plants. We also wanted to check if the if the carbon, nitrogen, and carbon to nitrogen ratio will be affected by nitrogen availability in environment. And we observed that pollen grains also store much more nitrogen inside compared to non-fertilized pollen grains. And it was similar in both species. And when we check the literature, carbon to nitrogen ratio was also related to activity, higher energy production, and this could explain why nitrogen deposition affect the germination ability of the pollen grains from the fertilized individuals. We also check if the metabolome is changed similarly to the seeds metabolome, and in this case, in both species, we observe differences in the metabolome. Some of them were upregulated, some were downregulated. To sum up our all observation, we can say that long-term fertilized plants, of course, will produce higher total biomass. They will produce seeds and pollen grains with much numerous production will be observed. The weight and size of seeds will be also higher, and we will have more nitrogen content inside. However, the quality of pollen grains and seed will decrease. Now we also know that the metabolite profile of metabolite can be, let's say, marker or molecular marker of the quality of the seeds in relation to nitrogen deposition and nitrogen availability for the plants. So thank you for your attention, and thank you to all of those who were participating in this research, and thank you.
Thank you very much for the interesting lecture. I would like to invite the next speaker, Marzena Guzicka. Personally, I don't like the winter season, but plants don't have the choice. That's why right now in the lecture, she will discuss whether the low temperature is an enemy or an ally for trees. and will outline the importance of the low temperature on the growth of trees, on the resilience of trees, and also on the long-term stability of population of trees. So the title is Winter, an Enemy and an Ally of Trees. The floor is yours, Marzenka.
Thank you, Eva. We are meeting in spring, probably the most beautiful season to visit Kórnik. But today I would like to talk about the winter, unfortunately. We usually think of winter as trees' opponent. Cold, frost, snow, all of this sounds rather not pleasant, but what if winter is not only an enemy? What if it's also an ally? It's question. Winter in our climate is very changing. We experience both freezing days and also period with temperatures above zero. Precipitation is often snow or frozen rain, but rain or select can also occur. Days are much shorter than nights, and winter brings several risks for trees. For example, sunlight warms the bark during the day, but temperatures drop rapidly at night, leading to tissue damage or bark and trunk cracks. Needles and branches lose water, but since the soil is frozen, roots cannot replenish it, leading to dehydration. May occur ice and snow damage, and repeating freezing and the frost pushes soil upward, exposing roots to cold air. And also important problem in winter is animals, because when food is hard to find, animals, for example, there feed on bark and young shoots. Winter but there is also something to protect trees. Snow. Snow works like a natural blanket. It protects plants, all plants, not only trees, from very low temperatures. When there is no snow, they are in more danger because root can freeze, plants can dry out, soil freezes and thaws many times. And under snow, the temperature is actually much more stable. Snow is also important in spring because melting snow provides water. Of course, snow is not always helpful because heavy and wet snow or ice can break branches and also bend them permanently. Sometimes snowfall is very intense, as you can see in this photo from cone collection. It was really hard work for our colleagues. Of course, young trees and seedlings are especially sensitive because their shoots are still very delicate. And another problem is cavitation. During winter, we have many troubles with bubbles. In winter, these air bubbles can form in the water transport system of trees, and this can block water flow and in extreme cases even lead to tree death, as you can see on this photo from our field experiment. Fortunately, in our climate, trees and plant development is synchronized with seasons. And it's really important because when cell division arrests and metabolic becomes very low, this time of hibernation is a positive effect for protection of trees, and different parts of tree become dormant, because dormant is a key strategy to allow us to survive unfavorable winter conditions. And as I said, different parts of tree become dormant, but buds are the most important because buds contain next year's shoots with meristematic tissue, with shoot apical meristem. And a bud is a small protected structure, and inside this structure there is embryonic shoot with an apical meristem, and this is stem mother cells for plants, and this is where future growth begins. And this structure is especially sensitive and especially need protection. Popular definition of dormancy say that this is absence of visible growth in all structure containing meristem. Growth stop, mitotic division arrest, and metabolic activity decreased dramatically. Different organs enter dormancy, but buds are especially important, as I said, because they contain this next year shoot. What I can say, dormancy is not static period, and it's not possible to give hard line, hard border between different stage of development during the year, because from active growth to ecodormancy is continuum. It's a dynamically changing period during all seasons, but it's not static. It's a continuum, and it's important because study of dormancy, because of that, are really difficult. Because we haven't good marker to identification of dormancy stages. We haven't good non-invasive marker, but when we are, it's key for all our analysis. And I repeat, it's once again, for initiation of endodormancy, we need shorter and shorter days. But for release of endodormancy, we need accumulation of chill units inside buds. But for release of ecodormancy next period, determinated only by ecological factors, we need accumulation heat units inside buds. And what is the next problem? At the same time, when we have dormancy inside bud, we have also phenomenon connected with acclimatization and frost tolerance, and it's really important. And this frost tolerance is not most higher during endodormancy, but in ecodormancy, in this period when true winter there is in our climate. And of course, dormancy is controlled at the molecular level, but dormancy is regulated by a molecular network similar to flowering, surprisingly, where flowering locus T acts as a growth promoter, while dormancy-associated Matchbox genes act as suppressor. Short days activating DAM genes to induce endodormancy, but winter chilling results in the downregulation of DAM genes and the upregulation of growth promoting factors that are necessary to break endodormancy. And also, in next step, ecodormancy initiation. DAM genes are highly expressed in dormant buds, and these genes are repressors which turn off FT to prevent too early growth. Also, plant hormones are very important, and especially ABA and gibberellins, because ABA keeps the buds dormant, and gibberellins helps restart growths. And this balance between ABA and gibberellins is crucial. And ABA also inducts dihydrins, very difficult pronunciation, dihydrins accumulation, and this protein protects cells from cold and dehydration by regulation of osmotic balance and also by preserve low metabolic activity during dormancy. And so we can say something quite surprising. Trees actually need cold. It's necessary. But there are also structural changes in buds. Cells can reduce communication with each other by closing connections via plasmodesmata. Is the basic for dormancy regulation. And in our research on spruce, we observed different ways of isolating cells, including complete disappearance of plasmodesmata, reduction in their number, and also blocking connections between cells by callus. And interestingly, unlike in birch or poplar, it was research of Rinne and van der Schoot. In spruce, callus persist also during ecodormancy and keeps isolation of the procambium and peripheral meristem. And we also observe differences in callus localization between these two different dormancy stages. And callus deposition is connected with developmental regulation, but not only, because it's also a response to cell damage when destructed cells are isolated by this way, as you can see on these micrographs in the corner. These structural changes are strongly, especially in ecodormancy. In my opinion, during spruce bud dormancy, key are changes in water status at first, callus accumulation at decomposition in different stage, and first of all, starch accumulation and redistribution. And it's obvious that structure is closely connected with function, and when we observe structure, we have also a lot of information about cells' activity. And for example, as you can see in these micrographs in the corner, during dormancy, cells are really active, and activity of organelles change often. And also, as you can see, chromatin is strongly condensed, but even in such stage of condensation of chromatin, we have transcriptional activity of nucleus. It's visible some areas with the condensed chromatin, and it's information for us that this part of chromatin are transcriptively active. It will be surprising for some people, I think. And but survival during winter depends not only on dormancy. This main strategy, but it's not enough, because we also need protect a meristem. But scales provide a physical barrier, giving meristem thermal safety, and they also act as a light filters, blocking mutagenic radiation, but not photosynthetically active light. Needles can to intensify this protection as an additional protective layer. And important parts of insulation this is the nodal diaphragms, isolation by anatomical way, gas below it, in gap, and this gap is also anatomical way for preserved of meristems, but also physical barrier. Important is symplastic isolation, and also isolation of vascular tissue between last year shoot and bud by tightly bound water. All these mechanisms work very well, and they evolved over a long time, but unfortunately climate change and may disturb the mechanism. Warm periods in winter can create a false spring, and buds may start preparing to grow too early and lose frost resistance. Also, warmer winters may cause the cold units necessary to break endodormancy, and these cold units not to be accumulated. The biggest danger comes when warm periods are followed by sudden frost. Buds that already started to develop can be easily damaged, and this can lead to serious ecological and also economic effects. So, is winter an enemy? Not exactly, because a proper winter actually helps trees. It provides necessary cold, increased frost resistance, prevent early growth, and in this sense, winter is an ally. To conclude, winter is a difficult time for trees, but also a very important one. Cold temperatures are not only harmful, they are necessary. Without proper winter conditions, trees may not grow correctly, and with climate change, this balance may be disturbed. Thank you for your attention. Sorry for technical problems, and I hope it won't snow, because spring in Poland is also really variable period. And I wish you nice weather and have a nice time in Kórnik. Thank you.
Thank you very much for this presentation. Now I would like to invite Professor Joanna Mucha. We all know that plants don't have the nervous system. However, the phenomenon of memory also exists in plants and in trees. And today we will find out what is the climate memory or ecological memory in trees and where it is located. I hope it will be possible to open the presentation. Oh yes, we have it. So
I would say so far so good.
So far so good. I hope no more technical problems will appear. So the title of the lecture is Rooted in the Past, Climate Memory in Tree Roots and Microbiomes.
Okay. Thank you very much for the introduction, and I also very grateful the organizer for opportunity to be here and present mostly my research. Okay, so Charles Darwin once suggested that the root tips act like a brain of a plant. Today, I like to take the idea one step further. What if roots also retain a memory of a past climates? And if so, how is that memory reflected in root threads and microbiomes? Forests are increasingly exposed to climate change, including higher temperature and more frequent drought. Here you can see the occurrence of the summer drought in 2018 and 2019, which was actually exceptional in the last 250 years. And of course, it impacts the growing season vegetation activities, which was definitely stronger than the European droughts in 2003. These changes affect not only above-ground processes, about which we can hear previously, but also what happens below ground, particularly in root and their interaction with microorganisms. Roots are actually not just an organ for water and nutrient uptake. They are a key interface between plants and the soil environment, mediating interaction with a wide range of microorganisms. Through their activities, roots strongly influence below-ground processes. And different species and even a population show contrasting strategies in how they grow, allocate resources, and interact with symbiotic fungi. This figure summarized two contrasting below-ground strategies in response to drought, as observed in beech and spruce. On the left, you have beech. On the right, you have spruce. On the left, we can see that the beech follows a strategy focused on rapid resources during the drought. It maintains active root growth, high mycorrhizal colonization, and produces short-lived absorptive fine roots. This allowed beech to quickly exploit available water and nutrients when conditions improve. In contrast, spruce adopt a more conservative strategy. What it means? Root activity is actually reduced. Roots become more protected and suberized, and resources are conserved rather than rapidly acquired. This reflects a strategy focused on survival and resource attention under stress. So these contrasting strategies show that tree species differ not only in how they respond to drought, but also how they regulate root activity and interaction with microorganisms. So it was my short introduction. So now we know that roots respond to both climate and environmental stresses. But we still know very little about how climate history can shape root-associated microorganisms, even when the trees grow under the same condition. So first, I would like to focus on the Scots pine. And this is a species with a wide range of distribution, which forces this species to adjust to different climatic conditions. And please have in mind, when I will be talking about the species, that these are studies on mature trees, because seedlings respond more strongly than mature trees. So, and just for quick reminding you about absorptive roots, because I will be talking about this. On the left you have a picture which has a typical anatomy of absorptive roots. This is a root which fully absorb water and nutrients, and also through this anatomy make an interaction with symbiotic fungi. So here, in these studies, we actually compare how Scots pine from different provenances allocate biomass to absorptive roots, which is important for the trees. And red lines show you how this population behave in common garden experiments, means when they are growing in the same environmental condition, but have a different origin. And what we can see that for the northern provenances, trees consistently invest more in fine absorptive roots, even when they grow in the environmental condition. Northern means that have a lower mean annual temperature, and you can actually see here that we have more absorptive roots and have the same pattern actually what we observe in latitude gradient. This suggests that root allocation patterns are actually strongly shaped by the climate of the origin and persist across environment. What is the consequences? Importantly, and still here in red you have how trees behave in common garden experiments. So importantly, this greater allocation to absorptive roots is linked to improve nutrient status in needle, particularly in nitrogen and phosphorus. So in other words, the more absorptive roots are, the more nitrogen concentration and phosphorus concentration we observe in the needle. So this indicates that below-ground threats directly influence above-ground function, enhancing nutrient acquisition. Here you have the results of another common garden experiments. Still on the left you have localization of the Scots pine population, which were grown in a Kórnik. And what can we see here? So we can observe, redundancy analysis and Permanova highlighted the significant influence of the seasonal variation, but more strongly by the tree origin actually for fungal community. And this analysis also shows strong association between mean annual temperature and precipitation and root metabolites such as carbohydrates and also here nitrogen with fungal community, a little bit less with bacteria. Beyond the composition, we also examine how microbes interact with each other, I mean across kingdom, and what we found is that the geographical origin of Pinus sylvestris strongly shape the structure and complexity of the fungal and bacterial co-occurrence network. So here I just show you only one of the region and which characterized, which actually the lowest precipitation. And what we can observe is the higher interaction, which we conclude from the number of edges, and also more highly dense interactive microbial community. So means that this cross-kingdom interaction also depends on the origin and shapes which the present, actually the origin shape the present microbe community and their and how they interact, which can influence ecosystem functioning. In another common garden experiments, here you can see also the location, the origin of the location of different population. We actually wanted to check how this climate influence not only below ground processes but also bacterial community. And what we could find that, for example, in spring, root chemistry is most influenced by the climate, and actually root associate bacteria. I mean bacteria which are inside the root show stronger response than soil bacteria. And these results showed that the climate origin influence root threads and also soil properties, which in turn shape bacterial community. It's not strong influence, but still we can observe it. Here we go a step further, so we actually would like to check how fungal community which are inside a root and in soil are shaped by the origin. So Scots pine provenance was a significant factor differentiating OTU level I mean community composition. And this suggests that the legacy of the climate is expressed through below ground environmental condition and plant and soil interaction. Importantly, provenance also influenced the functional composition of fungal communities. I'm sure that you know that ectomycorrhizal fungi help to roots to absorb water and nutrients, but saprotroph participate in making different resources available from organic matter. And So what we could observe here is that the relative abundance of key functional groups, such as ectomycorrhizal fungi and saprotroph, differ amongst origin. And here on this, you have it colored according to mean annual precipitation. So we could see the patterns. The proportion of soil saprotroph varied across communities associated with Scots pine provenance and was generally lower in trees from provenance with less precipitation. So this indicates that legacy effects extend beyond community structure and may also affect ecosystem processes such as nutrient cycling. To sum up this part, taken together, these results show that tree origin leaves a persistent imprint on root threads associated with microbiomes, linking past climate conditions to present day ecosystem functioning. In the second part of my presentation, I would like to focus for a while for environmental legacy effect. And I would like to tell you something about research, about watering and what happened after when we watering ecosystem after 5 years of drought. 5 years means that it's a long-term drought. And here you can see how different species react. Here we have BB as a beech, SS as a spruce. B mix means that it's beech root in mix place when root and beech intermingle, and S mix means that it's spruce in the zone when there were root and beech roots. And what we can, and here you have control plants in blue and plots experience previous drought as in red. So what we can see here, especially for the degree, we actually observed the delay of recovery, especially for spruce, and what we can see here, that the degree of mycorrhization is still far from this from control plots. Means that what's happened during the drought doesn't disappear when we rewater the plots. Looking at the functional groups, we see that the droughts also alters the relative abundance of key fungal types. So ectomycorrhizal fungi show greater stability, especially in beech, not for the spruce. And saprotrophic fungi also respond more dynamically. This demonstrates that environmental history, such as drought, can leave a persistent imprint on below ground system. And here, in the same experiments, we examine soil fungal community and how they respond after rewatering following, as I said, 5-year experimental drought. And what we can see here, that especially for saprotrophic fungi, we have a different community composition. And if we look at the different groups, we see still contrasting response, and it also can be seen in enzymatic activity. Enzymatic activity, which is actually important for organic matter degradation. And here you can see that this not only root zone, but also the treatment strongly influence enzymes which released nitrogen or carbon from organic matter. So taken these results together, these results show that rewatering doesn't erase the effect of droughts. Instead, fungal communities recover unevenly. And this legacy effect have clear consequences for ecosystem functioning. Of course, it can have actually implication for the forestry. That forest resilience depends on below ground climate memory, not only on current condition. And provenance selection influence also soil microbiomes and ecosystem recovery actually might influence. And what we could also see that mixing different species can buffer climate stress by stabilizing below ground interaction. So at this point, I would like to thank all collaborators involved in this work, including those whose project I contributed to. Together, we were able to gain a deeper insight into root microbiome interaction under changing environmental conditions. So I started with Darwin. So I would like to end up. So Darwin saw roots as a brain for a plant. I mean that they act like a brain for a plant. Today we might add that they also remember. And perhaps the future of forest depends how we manage that memory. So thank you very much.
Thank you. Thank you, Joanna. We came to the end of the session 1. I would like to thank all of the speakers, also to all of the participants for your attention. We didn't put a specific point in the program, which is discussion, but please remember that all the speakers are available during the coffee breaks and lunch. And if you have any question, if you are interested in any of the topics which were presented today, please contact a specific person and we will be happy to give you more information. Right now, I would like to invite you to the coffee break. And please remember to come back exactly at 11:40 for the next session. Thank you.
Okay. For the rest of you, and we will start in next minute.
Welcome everybody. After the coffee break.
Can I?
Yeah.
Okay, so we can start the second session, and our first speaker is ready to start. So let me introduce Tomasz Leski, and based on title, I think that Tomek will summarize, I don't know, 25 or even more research in our institute. So please.
Thank you for the introduction. But before I move to the presentation, so ectomycorrhizas in forest nurseries, I would like to present some details about the forests in Poland. Forests in Poland encompass almost 9.5 million hectares. This means that forests cover almost 30% of the Polish country. Around 77% of Poland's forests are managed by the state forests, and the annual timber harvest is in this area managed by the state forest amounts to around 38 million cubic meters. Consequently, there is an annual necessity to produce thousands of tree seedlings for the reforestation and afforestation the new areas. The state forests produce around about 700 million forest tree seedlings every year. This production takes place in forest nurseries. Currently, we have in Poland 385 forest nurseries. Most of them are traditional field nurseries producing seedlings with bare roots. Seedlings produced in forest nurseries must be of high quality to ensure the success of future generation and afforestation. There are many criteria that determine the quality of the seedlings produced in forest nurseries. Root system quality, shoot-to-root ratio, seedlings height, bud development, water potential of the seedlings, nutrient reserves, and so on. But one of the most important factors determining the success of future forest generation is properly developed mycorrhizal relationships. But what is it? Mycorrhiza. Mycorrhiza is defined as a mutualistic association between defined roots of plants and fungi in which both partners benefits from this alliance, as in this picture. In our climatic zone, most forest trees form symbiotic relationships with ectomycorrhizal fungi, namely so-called ectomycorrhizas. This mutualistic relationships is widely recognized as one of the most ecologically significant interaction in terrestrial ecosystems. It involves primarily fungi from the phyla Basidiomycota and Ascomycota, which colonize the fine roots of woody plants, such as, for example, Pinus, Picea, Quercus, Betula, Fagus, and others. The structural organization of ectomycorrhiza is highly specialized and optimized for efficient resource exchanges. Fungal hyphae form a dense outer sheet known as a fungal mantle that envelopes the root tips. From this mantle, hyphae penetrate the intercellular spaces on the root cortex, forming the network called Kórnik net. In forest nurseries, we can also find another type of mycorrhiza called endomycorrhiza. This type of mycorrhiza is essentially restricted to the plant genera from Pinus, and Larix. Endomycorrhizas have the same characteristics as ectomycorrhizas, mantle and Kórnik net, but also show intercellular penetration, as you see here in this picture, of the fungal hyphae into the living cells of the host roots. The functional basis of mycorrhizal symbiosis lies in a bidirectional exchange of resources. The host plants provides the fungal partner with photosynthetically derived carbohydrates, while the fungus enhances the plant access to the soil resources, mainly water and mineral nutrients. Through the development of extensive extrematical mycelium, ectomycorrhizal fungi significantly increase the absorptive surface area of the root system, enabling exploration of the soil volume far beyond the rhizosphere and root hairs. The absorptive surface area of the ectomycorrhizal mycelium can be up to 10,000 times larger than the absorptive area of the root hairs. Given the extremely important role of mycorrhiza in the life of trees and the particular in seedlings growth, we have undertaken a lot of years ago research into mycorrhizal association in Polish bare-root forest nurseries. Our research was aimed at, among others, verifying the beliefs that have been prevailing for many years that long-lasting bare-root nursery practices adversely affect beneficial microbiota. And especially that in forest nurseries, tree seedlings exhibit low levels of mycorrhizal colonization, and the diversity of mycorrhizal fungi present in the forest nurseries is limited. In our, as I said previously, in our studies we focus on the bare-root forest nurseries. Over the years, we have researched over 60 bare-root forest nurseries and the most important forest trees, such as Pinus, Picea abies, Larix decidua, Quercus, Fagus sylvatica, Tilia cordata, Betula pendula, and Carpinus betulus. Intensive nursery practices such as plowing, weeding, and root pruning make the production of spore caps of fungi in nurseries almost impossible. It is very uncommon to find the spore caps of fungal species such as Hebeloma, Laccaria, Suillus, or Telephora. Therefore, the only method for identification of the indigenous mycorrhizal fungi on seedlings produced in the forest nurseries is direct roots observing. Molecular techniques have greatly enhanced the ability to identify fungal species. The standard method in this type of studies involves isolation of the DNA, amplifying the ITS region, and the sequencing of using the Sanger sequencing. The ITS region is widely recognized as a barcode for fungal identification. In this way, we are able to identify fungi forming individual ectomycorrhiza that differ in appearance, namely so-called ectomycorrhizal morphotypes. Shortly we can assess the mycorrhizal association of tree seedlings in forest nurseries using the following key parameters: level of ectomycorrhizal colonization and richness of ectomycorrhizal fungal species. To our surprise, the level of mycorrhizal colonization in most of the studies nurseries and various tree species was very high. Very often, we observed complete colonization of fine roots by ectomycorrhizal fungi, and in many cases it was 100%. One example, this graph illustrates the level of mycorrhizal colonization of 1-year-old Scots pine seedlings from various forest nurseries. As can be seen, mycorrhizal colonization exceeded 90% in most cases. Only in one nursery, the colonization level was lower, but it still amounted to 85%. What about the number of the ectomycorrhizal fungal species? Once again, the results surprised us. Across all the nurseries studied, we identified 22 species of fungi on pine seedlings, 19 on spruce seedlings, 7 on larch seedlings. There were also 23 species associated with oaks, 26 with beech, 23 with linden, and 21 with birch, and 23 with hornbeam seedlings. Altogether, in total, in all the examined nurseries and on all the analyzed tree species, we detected 95 species of ectomycorrhizal fungi. But how does the species richness vary in individual nurseries? In the same nurseries for which I presented the level of colonization, the species richness of mycorrhizal fungi associated with pine seedlings ranged from 3 to 9, even 9 species on 1-year-old seedlings. Our research showed that one of the primary factors influencing the species diversity of ectomycorrhizal fungi in forest nurseries is the tree species, primarily whether it is coniferous or deciduous trees. Some fungal species are associated with both conifers and deciduous trees, while others are characteristic only for conifers or deciduous trees. For example, several species of fungi from genera Suillus and Rhizopogon are associated only with the pine. These distinct differences between conifers and deciduous species are also visible on this graph, which was created based on Jacquard species similarity coefficient. Another factor shaping the communities of ectomycorrhizal fungi in forest nurseries is the age of the seedlings. Research focused on Scots pine has demonstrated that while 1-year-old seedlings are often dominated by a single taxon from Ascomycota, this one, Wilcoxina micole-forming ectendomycorrhizas, 2-year-old seedlings support significantly more complex communities incorporating genera from Basidiomycota such as Suillus, Rhizopogon, Hebeloma, and Laccaria. 1-year-old seedlings was dominated by Ascomycota, 2-year-old seedlings was dominated by representatives of Basidiomycota. In case of oaks, dynamic changes in the species composition of mycorrhizal fungi and their quantitative relationships are observed even within the first 6 months after seed sowing. This is results from Martin studies. Initially, young seedlings are colonized exclusively with by the fungus Scleroderma. Later, other species appear, and among them, Hebeloma and Laccaria tortilis begin to dominate. Our studies of linden, birch, and hornbeam seedlings in 5 first nurseries have shown that location significantly impact the diversity of ectomycorrhizal fungal communities on the seedlings. This is likely due to the combination of environmental factors that are not fully defined in each location. Even in this same species, each nursery may host a different fungal species. Of course, there are many other factors that influence the number and type of ectomycorrhizal fungi present in the first nursery. This include also mineral and organic fertilization, irrigation, soil type and chemistry, fungal pathogens, presence of fungal pathogens, age of the first nursery, the sequence of the species of the over the years, and many others. In conclusion, we may say that ectomycorrhizal fungal communities in Polish first barrow nurseries are fairly diverse, and the level of colonization of seedlings is very high. And we may say that in general, artificial inoculation is not necessary in traditional first nursery practices, but may be used in the containerized nurseries. If you are interested in the issue of mycorrhiza in first nurseries, more details on these topics you can find in our publication and conference materials. Finally, I would like to thank all those involved in this study. Maria Rudawska, our chief, Marcin Pietras, Leszek Kaliński, Marta Kujawska, Daniel Janowski, Michał Iwański, and Lidia Trocha, and also special thanks to our friends from Lithuania, Algis Aučina. Thank you for your attention.
Thank you. Thank you for the perfect time of your presentation. And now we're going to stay in micro scale and below ground, because our next speaker is Leszek Karliński, who tells us that biomass of soil microorganism can be an indicator of environmental changes.
Gdzieś mi zawsze myszka tam uciekła. To jest ta prezentacja, tak? Podejrzewam, że... To jest to? Nie. Tutaj. Proszę. Powinno działać, nie? Działa?
Good day. So today I will try to briefly to talk something. Yeah, thank you. To show you briefly to talk something about biomass of soil microorganisms and why we use it, why it's important. So forest soil are among the most biologically active components of terrestrial ecosystem. They host diverse microbial communities that regulate vital processes such as organic matter decomposition, nutrient cycling, soil aggregation, and carbon sequestration. Therefore, soil microorganisms play a fundamental role in maintaining the productivity, resilience, and stability of forest ecosystem, but also the ecosystem of all the globe. Microbial biomass typically account for only about 1 to 5% of total soil organic carbon. This relatively small pool exerts disproportionately large influence on ecosystem functioning. Microbial biomass is widely recognized as a sensitive indicator of the biological status of soil. As microbial communities respond quickly to changes in the environmental conditions, microbial biomass is often used as an early indicator of soil disturbance and ecosystem change. So, estimating microbial biomass has therefore become an important tool in soil ecology. Various methodological approaches have been developed, including physiological, biochemical, and microscopic techniques. Today, I would like to focus on two biochemical methods that use specific indicators of living microbial biomass, namely ergosterol, linked with fungi, characteristic for fungi, and fatty acids, which offer much more indicator, much broader scope of indicators for different soil microorganisms group. And well, let's start from PLA phospholipids, characteristic for the cell membrane, are structural components of cell membranes and can be degraded rapidly after cell death. So this is a very good indicator of living biomass, living organisms. And NLFA, neutral phospholipids, are primarily storage lipids. So they reflect energy reserves, and in some groups, dormant or stress-related biomass. So they are not only the biomarkers of different microbial groups, but also the PLFA and LFA ratio can indicate the physiological status of the microbial community, active growth versus storage or as I said earlier, dormancy. And here is a such review of studies in which the authors use fatty acids as a biomarkers. So we can see this picture is very, very similar to the picture presented during the first presentation of Professor Dominik Tomaszewski. So most of studies are concentrated in the Europe, some in Asia, and in North America. So we can see that there are a lot of places where which are still waiting for studies. Sometimes, okay, we can say that we have some biomarkers of different microbial groups, but sometimes it's problem to divide different organismal groups because well the number of biomarkers are limited, and sometimes we can also support our studies using different selecting methods like meshbacks method, using usually meshbacks placed traps from fungal mycelium is placed in the soil. They are filled with sterilized and acidified quartz sand, And so this material, this sample is not attractive for instance, for saprotrophic fungi, but for mycorrhizal fungi, which are all-time connection with the sources of carbon, it's not a problem to overgrow. So this is a good method to divide, for instance, mycorrhizal fungi and mycorrhizal and saprotrophic fungi, which are the two main groups of functional groups of fungi. Well, fungi constitute a fundamental component of the soil microbial community, particularly in forest ecosystem, where they dominate key processes related to decomposition of complex organic matter, regulation of the carbon cycling and storage, stabilization of soil structure, support plant nutrition, mycorrhiza, and control nitrogen availability and separation of soil-borne pathogens. So it's a very important group. This is the second group. The second group are so it's a saprotrophic fungi, which are typically are selected fast growth, rapid. The characteristic for them is rapid resource exploitation, high metabolic flexibility. We can say that saprotrophic and mycorrhizal fungi form a functionally coupled but competitive continuum. Saprotrophs release nutrient through decomposition, whereas mycorrhizal fungi intercept, immobilize, and channel these resources into plant fungal networks. Their interaction is a primary regulator of carbon sequestration, nutrient cycling, and ecosystem stability in terrestrial system. Okay, thank you. And here is one example of interaction of the example of the concurrence between mycorrhizal fungi and saprotrophic fungi. This is the negative correlation between external mycelium of ectomycorrhizal fungi production and the presence of woody debris and the study sites. Woody debris, which are a source of food for saprotrophic fungi. The second example is also negative correlation with the presence with the number of saprotroph sporocarps at the sites. And in such a situation, as I previously said, very useful method for dividing these two groups is our meshbacks, which we use for many years, and it's working very well. So as a key structural component of mycorrhizal symbiosis is a very well developed external mycelium, which penetrates the soil solution and accounts for 65 to 80% of total mycorrhizal fungal biomass in soil. By greatly expanding the root system, absorptive surface area, this mycelium plays a crucial role in acquiring and transporting water and nutrients to plants. It also contributes to soil forming processes, facilitates mycorrhizal colonization of fine roots and links plant of some species to enable circulation of carbon and nitrogen compounds between them. And we can also, despite the significant role of external mycelium play in forest ecosystems and their potential as an indicator of environmental change, relatively little is known about mycelial production in forest soil and the factors influencing it. And most studies up to now are concentrating in the northern part of the Europe. Some examples of studies we can find in a central or in southwest part of Europe and United States. In Poland, we were working with different clones of poplars, Scots pine or PCA abies in mountains and in the lowland areas. Well, and soil moisture. Soil moisture plays a crucial role in shaping extraradical mycelium biomass. Water deficit in soil can be more detrimental than, for example, contamination with heavy metals, to which fungi, especially under long-term exposure, may exhibit considerable tolerance and adaptive capacity. Also, the conversely, prolonged flooding has a distinctly negative effect, especially for ectomycorrhizal fungi. To less extent, we observe it for arbuscular fungi. Temperature and moisture generally support biomass production. The development of external mycelium is generally favored by lighter, well-drained soils rather than heavy soils with low oxygen availability. Increasing depth and associated reduced availability of nutrients and oxygen are better tolerated by arbuscular fungi than ectomycorrhizal fungi, what is very well visible in case of tree species like poplar or willows, which create mycorrhizal association with both groups. Also, soil fertilization are important factor influencing both the production of external mycelium biomass and the conditions of the plant-fungus symbiosis. Nitrogen levels of moderate nitrogen availability may stimulate production of mycelium, but excessive nitrogen deposition in soil can reduce biomass for over 50 to 80%. So the same we can see in the case of litter thickness and fertilization. Fungal biomass relates also to plant cover, host tree effect. Differences between tree genotypes are particularly pronounced under also stress conditions, but the fungal external mycelium biomass reflected very well these differences. We also saw the connection between the growth parameter, the photosynthetic apparatus and the biomass in rhizosphere, but also for external mycelium biomass. So bigger trees are okay. Yeah, so hurry up. And also the next factor is also the age, which influences. In case of coniferous, the production of mycelium up to 20 years is growing. Later, the production is we observe stabilization of and decrease of the production. A bit different picture we can see for instance, for poplars, which is connected with carbon allocation strategies. So at first, most of carbon is invested in building the mycelium net. Later, more carbon goes to the production of sporocarps and different survival structures. The same, we have seasonal differences in biomass production. Also lower for values for winter and spring season, higher for summer, autumn seasons. Okay, and very valuable factor is the ratio of fungal to bacterial biomass, which is indicator of a direct or inorganic matter transformation. So these values are higher in coniferous forests, and in deciduous it's varied about 1 to 3. And the lower value we can observe for agricultural lands. And there are some tendencies which we observe for so. Okay, and also important player in transformation of organic matter in soil are bacteria. But in the Central European forests, they are more they play a more complementary function to that of fungi, and we can see that there are some groups of decomposers, nitrogen bacteria responsible for nitrogen and phosphorus transformation and circulation, as well bacteria which supports suppress pathogens and etc. And also we can divide bacteria according to the cell wall structure or gram negative, gram positive, gram negative bacteria. Gram negative, more abundant in deeper soil layers under stress conditions, high resistance to desiccation, nutrient limitations, and gram negative with thin cell walls, dominated mainly in rhizosphere, carbon rich. And the ratio of biomass of gram positive, gram negative bacteria differences in soil have a clear ecological and functional basis, largely determined by their cell structure, with a thick peptidoglycan layers in G+ and a thinner layer together with the outer membrane containing lipopolysaccharides in G-. These structural differences translate into the distinct life strategies and responses to environmental gradients. So in here also Some examples, we also observe fluctuation, seasonal fluctuation in the values of this ratio. And here is also an example, the impact of soy depth. With soy depth increase the ratio of gram plus bacteria. Okay, so I concluded to the end, so I try to the most dense summary. This presentation was only a brief overview of the relationship that can be explored using specific indicators of soil microbial biomass. However, it's important to remember that the more research techniques we apply, including chemical, molecular analysis, and traditional methods based on the direct observations, the more complete will be our understanding of complex soil processes. Yeah, and then we can, at this moment, I can stop. So thank you for the attention. I hope this was not too long. Thank you very much.
Thank you, Leszek, for a nice presentation. We are sorry for these 2 yellow cards that you probably didn't see. And our next speaker is Marta Kujawska from the same department. And Marta have a speech about Scotch broom, the plant that I get to know what is it, what is the Polish and Latin name today morning. And Marta tell us about the secret connection between Scotch broom and fungi, symbiotic fungi.
Okay, hello everyone. Yes, my presentation will be focusing on Scotch broom. I know that many studies always focus on pine spruce, deciduous trees, but today I would like to focusing on neighborhoods, and this is the Scotch broom is a native neighborhood, but it's quite dangerous. And why I try to ask you and explain in my presentation. This plant, Scotch broom, maybe it's not popular, like Marcin said, but it's commonly associated with several things in the world. And first of all, yellows, beautiful flowers. And in traditional use, broom is the things historically using to clean the floor. So the people use these steams, and the broom is changed the name. In the ecological and forest aspects, is associated with soil acidification and, of course, nitrogen fixing ability. So it's very important and highlights their invasive character. And few words about these beautiful flowers. Here we can see the typical flower structure of Scotch broom as an example of Fabaceae family. These flowers have a very characteristic shape, which resembles a butterfly. It consists of 3 main parts. The large upper called the standard, 2 in the middle as named as wings, and the lowest that is together and form the keel. And in this keel is a very important location because there we can find stems and pistils which protect and play very important role in pollination. So this place is highly adapted for these very important insects like bees. And when a pollinator lands on the flower, it presses down on the keel and can realize of pollen onto the insect's body. So this is a very specific mechanism, increases pollination efficiency and helps ensure successful reproduction of this quite dangerous plant. Few words about distribution. Like I say, it's a native plants for Europe, but it's very invasive species in the whole world, in Australia, New Zealand, and also in America. By the Scots broom is very often caused by a wildfire. And why is dangerous? Because it's very protected by higher temperature, and their seeds are also tolerant low temperatures. So the expansions of this species is quite easy. Additionally, as a Fabaceae species, it forms symbiosis with nitrogen-fixing bacteria. So for this, these plants colonize nutrient-poor environments. So when Scotch broom live together with another plants, another trees, there is for the first place, and this is a winner. Here you are shrubs growing before flowering, so maybe it's not beautiful, but this is a normal picture. They're growing always along forest path because they love light, and you can't find these shrubs in the middle of the forest. They love light. The invasive success of the species is driven not only by their biological traits, but also by interaction with microorganisms. In the middle, you can see the stems that is even green. So also it's helpful for the species to persist in different regions and in different temperatures. Additionally, on the last picture, you can see seed coats, long-lived seed coats allowing seeds to remain viable in the soil and create a persistent, very huge seed bank. Much of the species' success as an invader is not just due to its own traits, but this interaction. And I am focusing today on not only on fungi interaction, but also on nitrogen-fixing bacteria and allelopathic compounds. But these three pathways cause that these shrubs, this Scotch broom, is very active in the surrounding soil environment and changing nutrient availability. That is direct impact, direct influence on surrounding plant communities. In the previous slides, I pointed out the success of the broom is closely linked to microbial partners. And here I would like to say more about this bacteria. So look at these fixes that between 20 and 100 kilo nitrogen per hectare per year they can add to the soil. That is a really huge amount of nitrogen, but it's also very dangerous. This enriches not only fuels broom growth and develop, but also reshapes soil chemistry, not always in positive aspects. And here I would like to say more about these changes in the soil. Scotch broom changes the soil chemistry and fungal interaction because this is a very close connection. In this paper of Slezak's was show how Scotch broom influence on Douglas fir fungal communities. So, because Scotch broom add large amount of nitrogen to the soil throughout their root nodules, this increases total soil nitrogen, changes the balance between other nutrients. And according to these authors, nitrogen levels rise after broom invasion, but also this level rises is stable and stay also after removal these shrubs. This increases nitrogen level rise is also form the shifts over time. Right after invasions on removal, nitrate that is in the middle can increase also due to the mineralization and nitrification, but later nitrogen often declines, not because nitrogen disappears, because we have this nitrogen in a huge amounts, but because nitrogens leach out of the soil profile and acidifies the soil. Additionally, the balanced nutrient is also touched by other elements like calcium, magnesium, and phosphorus. So this chemical shifts cause reduction ectomycorrhizal colonization of the main host in these places, and in this case of Douglas fir. I would like to focus on two very important mechanisms that drive this reduction of colonization, fungal colonization. We have to remember about allelopathic compounds in this case from broom litter, and of course on a nutrient imbalance that reduce the tree's dependence on the fungal partners. Maybe you don't know, but the Douglas fir and many coniferous tree species are obligatory connected with ectomycorrhizal species. So this problem that the broom and their absence cause a lot of changes in the soil chemistry, this also influence on a regeneration, on a develop of Douglas fir, and these trees also reduced forest productivity. In another example, I would like to show you not only what happens to the soil, because we know that we have many changes in soil chemistry, but how these changes change also the living networks in there, and how this microbial community shifts. So what is the most important? Nitrogen enrichment reduced plant dependence on mycorrhizas. This is really not good when the main host species lose of them ectomycorrhizal specialists. And these authors also shows that ectomycorrhizal abundance and a community composition shifts that also for this reason mycorrhizal communities is slow and variable due to the spore longevity and dispersal limits, because the seeds and the organic matter of broom are very long time in the soil. And the last example I would like to show you is experimental pot experiment where the authors want to isolate the soil legacy of Scotch broom in the natural conditions, but also in experimental conditions. Growing Douglas fir was slow. The invited soil was the main reason of this reducing seedlings grow. So we can say that provide direct experimental proof that broom leaves behind a harmful soil legacy that suppress Douglas fir development, regeneration even after the invader is here. And a little bit information about my studies and my work with Scotch broom. Here you can see the basic arbuscular structure that are in Scotch broom roots. On the first picture by A is arbuscule, so this is very important, the most important structure of this type of mycorrhiza. And we checked how change the colonization by arbuscular mycorrhiza in Scotch broom roots in 3 different times, before flowering, during flowering, and after flowering. And the lowest proportion of total arbuscular fungi was recorded after flowering, and this difference was significant compared to the proportion both before and during the flowering. This is easy because during flowering and before flowering, this mechanism, this time is very cost for plants. So a lot of nutrients, a lot of changes, a lot of energy plants need, and the fungi help in them. And the highest proportion of arbuscules was recorded during flowering, so this is a source of energy, and the lowest after flowering. Generally, working with arbuscular mycorrhiza is not easy because very often we can't find and we can't see this arbuscule. So work with roots Scotch broom was really present for me. And in the conclusion, management implication, maybe it sounds like a mission impossible, but I know that this microbial interreaction create very, very long-lasting soil legacy effect. So it's not easy to pretend the forest before the consequences. But maybe we can, in these processes, help explain why Scotch broom invasion and why can reduce long-term forest productivity and complicates restoration efforts. Maybe in Europe, Scotch broom is not a problem, but in America, in Australia, where wildfire are very, very common, is a really big trouble. And thank you for your attention. If we want more details about Scotch broom and about work with mycorrhiza, you can find information about Citicius coparius in this our last articles. And I will be with you to Sunday. So maybe if we want to have a question or want more information, I'll be with you. Thank you.
Thank you, Marta. Thank you, Marta, for presentation. And now we switch to the biochemical and omics research. And let me introduce Agnieszka Szuba, the leading scientist in omics method in our institute.
Yeah, but give me a minute because, okay.
Okay, I will talk. No problem. And today, Agnieszka tell us about the how we can understand tree physiology function based on omics analysis and biochemical analysis, please.
Okay, ladies and gentlemen, I really love omics, so today I will present you a little bit more technical presentation. Can I take it? No. Yes? Okay. So I will show you how to use biochemical and omics, especially not targeted high throughput MS analysis during the field study. I will show you challenges and some possibilities, and I will use as examples. I will use our own data to show you somehow first-handed and practical information. So, as a scientist, we are interested the most how plants react to harsh environments. Various factors could create stress in plants, and each factor will create a particular specific risk for plant, and plants will specifically respond to each factor. But even if we're talking about the same stress factor, the response could be different. We'll observe something quite different when we will analyze acute stress response, and then when we analyzed plants which are already tolerant or adapted to stress. So plant stress response is very complex and hard to analyze. Maybe even more important is that only small part of this response could be detected with phenotypic observation. And all points not pointed here with red lines will be not visible without using biochemistry and chemical tools. So for us, if you want to really know the level of stress or level of tolerance, it's really important to use these biochemical tools. But when we're working in the forest, it could be a really big challenge. This is because plants growing in controlled conditions behave quite differently than those which are growing in natural conditions. And this is important because majority of plant stress response is analyzed in controlled condition, and when we're talking about the most advanced techniques, we are talking about analysis frequently done not only on whole plants, but sometimes on the individual cells. That means that the knowledge about stress response, and we know a lot about abiotic stress response, is the knowledge about response of plants growing in highly artificial conditions. And this is because one of our goals was to use those techniques in the field condition. But when we're working in forests, even simple sample collection could be a big challenge. When we want to analyze some biometrical data, we could just simply collect and well-described material, it's enough. But when we want to analyze biochemical and especially omic data, we must to preserve our material immediately at the low temperature, either in liquid nitrogen, even if we're working in the middle of the forests. Next, when we're working with roots, for example, we need to clean our samples because we just don't want to analyze contaminated samples. Yes, soil contains a lot of proteins, a lot of compounds, so we really need to wash roots with icy cold water also in the middle of the forest, yes? And equally important is to get rid of this water because otherwise we'll have samples diluted with ice. And how important this step is, we know since we analyzed data from one of our experiments when this step was not carefully done for several samples. As you can see here, this fact has really huge consequences. We cannot analyze several variants. Okay. the next problem is that the structure of plants growing in nature is quite different than those of growing in vitro or controlled condition. Those plants have thicker cell walls, are covered with waxes, have huge amounts of secondary metabolites, and all those facts have really significant consequences. Usually we need to use really much more complicated extraction methods. Yes, it's especially important when we're talking about proteins. But probably the most important things that we cannot control environments. That means that each individual plant's growing slightly different condition. And of course, it's really important. Sometimes we compare variants which are located hundreds of kilometers away. That means that we compare variants, plants not growing even in various conditions, but even in various climates. So it obviously have a huge consequences when we're drawing our conclusions, yes? Even when plants growing next to each other. Oh, this is our director, you know? This is Professor Jagodziński here on this picture. So even if plants growing near to each other, that it doesn't mean that they are growing in the same environment. Then we're talking about so-called microenvironments. And this is also really important because it really affects the final variation within a particular group. So final variation in omics study, and it will affect power of our statistical tests. Yeah. So this brings us to the next point. The challenge is not ending when with the sample collection itself. The really next important step is to compose our sample. The sample which we use for the extraction, yes, must truly represent our plants, but also we need to minimize this effect of microenvironments as much as possible, of course, when we are not interested in those microenvironments itself, yes. Sometimes a good idea is to use so-called pooled sample, when several plants are mix each other, yes, but sometimes not. Still, the decision how to compose our samples is one of the key ones. It must be really well planned and sometimes even optimized. In some cases, we will use sample fractionation. That means that from one sample, we will obtain a much more information, but it also that means that at the final level, we'll analyze by MS much higher number of samples. Sometimes the preparation of final sample is really time-consuming. Here, for example, I show you cell wall fractionations, and hydrolysis of cell wall fraction could take several days. Usually, when we think about high-throughput MS analysis, we're thinking about comparative analysis of hundreds of compounds. Yes, but sometimes we are interested in few particular compounds. Then we could analyze, perform quantitative study. But for this goal, we need to build our calibration curves. And all of that, using fractionation, calibration curves, and so on, will increase number of samples analyzing by MS. And it's important not only because omics studies are still expensive, but also because we're dealing with so-called batch effect. That means that spectrometers not always acting in the same way. We'll see small differences in retention times. Yes, we'll see also, for example, degradation of the sample when it will be stored long-term in the autosampler. All those facts will, at the end, will have the results with mass spectra will slightly differ between each other. And that means that we'll need use a really complicated data analysis will be also harder. Yes, we'll need some different normalization methods and so on and so on. So it's really good to really rethink our goals and to plan our study in our optimal ways. Not always much sample is a better idea, yeah? And obviously, we're working with living organisms. That means that we're working with organisms made from millions of compounds. And we are able only identify only part of them. Actually, we are only able to identify those compounds which mass spectra are already stored in databases and which mass spectra may be compared with data which we obtained during our studies. That means that majority of our data will remain unidentified. Okay, this still means that we are able to identify hundreds or even thousands of compounds. Such kind of results are not possible to obtain during, for example, biochemical study when we analyze level of chlorophyll or level of proline, not mentioned to phenotypic observation. But still we are limited. But in the same time, as I said, we could analyze hundreds or thousands of compounds. And we could really detect those compounds which truly different between variants. And this is important because they are not always those compounds which we plan to analyze with targeted study. Yeah? Moreover, let me repeat, we only could identify those compounds which are deposited in databases. And this is important because, as you can see here, our extracts contain thousands of compounds. Today even, we are able to identify 10 to 20% of those compounds. And when we're talking about proteomics, the situation is even worse because then the species is important, and when we're working with not model plants like trees, the level of identification drop even more significantly. Okay. I will show you a lot of limitation and a lot of challenges, but actually which kind of results we could obtain during those research. Obviously, we could analyze hundreds of compounds at the same time. That means that using PCA, we could detect general similarities and differences between variants. Using ANOVA or t-test, we could identify all compounds which differ between variants, and moreover, we could rank them, for example, using fold change ratio and other aspects. So we'll have a lot of information. With this huge amount of information, we could analyze many processes at the same time. That means that we could answer the question how and why. For example, here, this is just an example. Here, this is an analysis showing how ectomycorrhiza affect heavy metals plant stress response. And today, we could use a number of online platforms which allow us to identify and analyze data even in more sophisticated, holistic view. That means that we could find some interaction will be most probably omitted without those tools. Here, also an example showing that we found a huge role of ribosomes and endoplasmic reticulum in the plants adapted to soil. Most probably we will not find this information without those tools. And actually, basically, that's it. And finally, I would like to show you a few particular examples on which question could be answered using those tools during the field study. For example, we use omics and biochemical techniques for selection the best cultivar. We select the best plants, that means that the most tolerant plant for bioremediation of industrial waste landfills. And we use also biochemical and omic study to monitor not only soil condition, but also the true tolerance level in those plants. We also analyze which tolerance mechanisms were the most important here at the secondary on the picture. That was the most important for the survival of trees where they are really growing in the contaminated soil. We analyze, for example, naturally growing adult widows affected by sudden unexpected flood, which highly contaminated, water highly contaminated with arsenic. Those trees react really differently, and we just wanted to know why. We analyze also mechanisms of acclimatization. That means that we analyze trees which were affected by stress condition for many, many years. And this is important because there are still just a few examples of using omics techniques for the long stress trees. And quite recently, we used metabolomics tool to analyze effects of transposition population to a new environment. And even if we didn't detect any biometrical changes, we detect that those populations are not fully acclimatized to a new environment. And this is, of course, important, very important in context of current climatic changes. And sometimes biochemistry could also show us no stress, even if we clearly expected it. For example, the tiled biochemical study of free seedlings growing naturally in extremely harsh environment of current active query site, and we don't detect any signals of oxidative stress. Those trees, seedlings, were not stressed. Look, where they're growing, yeah. So in fact, there is more and more information that plant response of plants growing in control condition could be quite different than the response of plants growing in nature when they are part of complex ecosystems. And this is because, and this is the fact, and this is because we want to use those techniques as frequently as possible. And with this conclusion, I will thank you for your attention. And I think that I'm on time.
Yes. Thank you for perfect presentation and very nice interaction with audience. So now we jump on lianas, because our next speaker is Olena Blinkova, who tell us about multi-level response of urban forest lianas along environmental gradients. Please.
Nie ma.
A tutaj nie.
O, jest. Jest. Laski.
Jest.
No, teraz jeszcze jest, okej.
Thank you, Martin. Good afternoon, dear colleagues. Today I will like to present our research entitled Multi-Level Response of Urban Forests, Lianas along Environmental Gradients. The central idea of this presentation is that lianas in urban forests are not only structural or passive components of forest-urban vegetation, they also respond to local microhabitats gradients. I will focus on Hedera helix, or common ivy, as a model species, and I will show how it responds from shoot morphology to functions, hydration, pigment traits, and biochemical traits. To begin, I will briefly explain why forest-urban ecosystem are very particularly interesting system for this type of research. Urban forests are highly heterogeneous systems. Within a small area, they differ in air temperature, local light availability, soil moisture, thank you, soil temperature, and disturbance density. These different traits create a mosaic of microhabitats where vegetation develops under combined impact of local sites, condition, and urban disturbance. Also, urban forests are ecologically important because they support biodiversity, alpha, gamma, beta biodiversity, and contribute to local regulation of air temperature, water balance, and other environmental gradients. Therefore, urban forest ecosystem suitable systems for studying trait variation and plant response of environmental heterogeneous. Plant functional traits help us to understand how species respond to environmental limitation. Traits such as leaf area, specific leaf area, biomass allocation, pigment concentrations, and biochemical traits reflect different ecological strategies. Some traits links to rapid resource acquisition, while others indicate resource conservation and stress tolerance. So this is strongly related to the concept of the leaf economicum spectrum. For lianas, functional traits are particularly important. Why? Because this life form plays an important role in function forest ecosystem and occur in a forest across different climate zones of the world. And second, lianas contribute the forest structure and dynamics. So by studying traits, we can understand not only how plant looks, but also how it functions under different microhabitat conditions. Hedera helix, or common ivy, is a perennial evergreen liana from the Ferrariaceae. This species has a native range spanning the Mediterranean basin, Western, Central, and Eastern Europe. This species is especially useful model for. Why? Because it combines several important features. First, it's shade tolerant and ecologically plastic. Second, species tolerates a broad range of soil conditions. And third, this species has different shoot types, vegetative and generative climbing shoots. And these differences of shoot types is very important for our study because vegetative and generative shoots may not respond to environmental gradients the same way. So at the same time, despite its natural presence in temperate forest, ivy is increasingly common in urban green areas where microclimatic environmental gradients and disturbance intensity shape its local population structure and other parameters of local populations. The main objective of our study was to assess how environmental gradient structure, multi-level trait response of common ivy in urban microhabitats. The key point here is the word multi-level. We did not analyze only one trait or only one level of biological organization. We combined morphometric functions, hydration, pigment traits, and biochemical indicators. So we tested three main working hypotheses. First, we expected that vegetative and generative shoots, less healthy and damaged leaves, would differ in their trait profiles. Second, we expected that limited environmental parameters for our model species, such as soil moisture and light variability, to be a main environmental drivers structuring multi-level trait response in our model forest conditions. And third, we expected that vegetative shoots to show stronger coordination matrix groups and higher integrative performance than climbing generative shoots. So our research was developed in 3 stages. In third stage, we focused on morphometric and functional leaf response, and the results showed that limiting environmental parameters of our model species, such as light variability and soil moisture, were key drivers of variability in vegetative shoots of common ivy. In second stage, we focused on functional adjustment and water status. We analyzed above ground biomass allocation, leaf and stem water content, and above ground biomass water content. At the third stage, expanded the framework. We included both vegetative and generative shoots, always healthy and damaged leaves. And this expanded our single framework, and we combined morphometric functions, hydration, pigment traits, and biochemical traits. So, Yes. So the logic was gradual from morphology and variation to functions and hydration, and finally to multi-level integration. Our study was conducted in wooded areas, including botanical and dendrological park, urban forest habitats. These sites represent different structural and other local environmental parameters. They differ in canopy opens, light variability, soil moisture, and disturbance intensity. The key point here is that we did not treat urban forest as a uniform habitat. We considered it as a set of microhabitats organized along gradual environmental gradients. This has allowed us to connect plant traits with real environmental variables and to find spatial scale. In the beginning, we analyzed morphology and variability. Then we added functions and hydration traits to understand adjustment mechanism. And finally, we integrated different traits to multi-level framework. The final step was the development of ecological index of the additive response in Hedera helix. So main results. In first stage, we focused on environmental drivers of trait variation in vegetative shoots. Environment conditions were assessed using Ellenberg indicators values, such as moisture variability, soil reaction, salinity regime, and other parameters. Also, we analyzed local population quality index, and the results showed that common eye local population occurred mainly under mesophytic conditions. Daily light integral light variability was one of the most important factors of the fine spatial scale, and at the same time, water status influenced the vitality and structural variability of local population and other parameters of our model species. Here we can see that local population differ in the vitality structure and relationship between local population types and daily light integral, depressive, equilibrium, and prosperous. Limb functional traits, such as specific leaf area, varied along the studied light gradient, and leaf structure reflected local environmental conditions. So, common ivy adjusted its functional strategy according to leaf economical spectrum, according to limiting environmental parameters for this study, such as light variability and soil moisture. The second stage focused on leaf water status and Ellenberg indicator values. The results show that leaf water content is a sensitive parameter of soil moisture, even under general mesophytic conditions. It is very important because hydration traits, hydration status can reveal stress or limitation before more visible changes in plant structure appear. So both fresh and dry mass declined with light variability, and this is indicated about reduced above ground biomass under higher light variability. And this higher light variability may act as a stress factor for our ivy. At the same time, biomass allocation reflects combined impact of light variability and volumetric water content. The second stage demonstrated that increasing of daily light integral was associated with changes in hydration status. For example, leaf water content, stem water content, and above ground water content responded differently. The third stage was the most integrative part of our research because we analyzed impact of volumetric water content, electrical conductivity, and soil parameters, soil temperature on the functional leaf traits shoots, vegetative and generative shoots, as well as healthy and damaged leaves. The results showed that leaf damage was associated with reduced leaf functional traits, such as specific leaf area and leaf mass fraction, particularly only in vegetative shoots. Vegetative shoots showed structural differentiation under low soil parameters, volumetric water conditions, but in the same time, healthy leaf on generative shoots exhibited higher specific leaf area, leaf mass fraction, at intermediate level to high level volumetric water content. So PCA summarizes the relationship between among functional traits, environmental variables, shoot type, and leaf condition. The results show that healthy leaves of generative shoots were positioned at positive PC1 values and associated with higher soil temperature and electrical conductivity. But damaged leaves of generative shoots clustered low PC1 and PC2 values and associated with higher leaf mass area. Orientation of specific leaf area relative to leaf area, soil temperature, and electrical conductivity in their inverse relationship. Impact of lac intensity, daily light integral on the functional leaf traits of common ivy shoots. The results show that pigment content showed a non-linear response to daily light integral, for example, to water content or other environmental gradients. Pigment concentration decreased, but carrots declined more gradually, and damaged leaves of generative and vegetative shoots has low pigment concentration. At the same time, healthy leaves on generative showed the highest pigment levels at intermediate daily light integrals. Why? It is very important because it showed that the higher daily light integral has become a stress factor for ivy, especially when it combines water limitation and leaf damage. The strongest environmental contributors for pigment traits of ivy leaves were soil parameters, soil temperature and volumetric water content, and light variability. Especially daily light integral showed a negative association with all pigment concentration, and water content was the main positive predictor of pigment concentration. But carrots associated only with soil parameters such as electrical conductivity and volumetric water content, but not with temperature soil. So, this volumetric water content was most strongly associated with leaf, stem, and biomass hydration in vegetative shoots than in generative, and soil temperature showed positive relationship with stem water content. Electrical conductivity exhibited a limited effect with positive association mainly in damaged leaves on generative shoots. Overall, this is very important because hydration status, hydration response depend not only on environmental but also on shoot type or leaf damage. Protein contact in ivy leaves differentiated among functional groups too. For example, lowest and most stable levels were established in healthy leaves of vegetative shoots, but highest values and greatest variability were established in damaged leaves or generative shoots. It is very important, but this result shows no significant relationships with measured abiotic factors. The integrative ecological response was developed to summarize adaptive response of our model species across several biological scales. Index differentiated morphofunctional groups such as healthy and damaged leaves, vegetative and generative shoots. And we see that optimal values were established in healthy leaves of vegetative shoots, but the lowest performance was established in damaged leaves of degenerative shoots. Greatest variability observed in generative shoots, healthy and damaged leaves. So the main conclusion is that environmental gradients for our model species Hedera helix, limiting environmental parameters, soil moisture and light variability, structure tray variability in urban forest lianas, especially local populations. Liana responses are expressed through coordinated change in different traits in several levels of biological organization. In common eye with these gradients differentiate shoot type, while leaf damage increases trait variability. In the same time, lianas should not be viewed only a passive component of urban forest vegetation. They are functional agents that interact with canopy structure, modify understory condition, and respond rapidly to changes in light and water regimes. So extension to other liana species, future research, and study across latitude gradient, cities, seasons, different types of forest, and vertical forest gradients is required. Lianas handle urban stress very well, but their response is not very simple. They are coordinated, multi-level, and strongly dependent on the local microhabitats. Thank you very much for your attention.
Thank you, Olena, for your talk. And the next speaker, the last one in this session, is Mikołaj Wawrzyniak, who tell us that every seed matters because seed science has a great history in our institute. And the presentation will be about importance for forest practice. So yes. The floor is yours.
Yeah, so welcome everyone. My name is Mikołaj, and the good news is that I am the last person before the lunch break, so please bear with me just a little bit longer. I will talk about seeds in forestry, and I don't know if any of you works with seed. I guess that some of you, but I'm also certain that all of you know how important seeds are for our society, as 70% of calories that we daily, as a humanity, use for our organisms to run comes from seeds or seeds-derived product. So seeds are very important structures from plants that we use for our benefits, not only in agriculture, but also in forestry. Because every tree, every forest starts with a seed, and it's always a beginning. So knowing seed biology and knowing seed physiology can help us use them in the most efficient way for our benefit, but not only ours, but also for ecosystem and the nature. So when we think about the seeds, in my presentation, we could focus on two important paths that overlap a lot, because we will think about forestry production, but we also will think how to use them as a method for preserving and conservate biodiversity. So few things to start that we can keep in our mind, that we don't collect all seeds that there are. Many of seeds stays with the tree. They are used as a natural resources for regeneration. We need to remember about the seed biology, the seed physiology, to use them more efficiently. So we need to know what conditions are good for storage, because we don't want to store seeds that won't germinate or make them less viable. The idea of the storage is to keep the viability of the seed for as long as we can. The success of the storage of dormancy release and then the germination determines our outcomes, if this process will be a success or not. Trees in the production, seeds in the production, that's one thing, and then also this is also important to keep them safe for ecosystem regeneration processes, projects, and to help save as much biodiversity as we can. Seeds are a perfect organ for such task and to work with them like this because they are biologically made to last. There's a lot of traits that we can use that helps us preserve them. Here we can think a little bit why we will store the seeds. And also here we can think in two ways, productionally and in the production way and in the conservation way. In production we store the seeds because trees very often have this characteristic that the masting years, they are very erratic. Now they change. There's a lot of studies coming in how the climate impact the seed production by trees. But in general, in previous year, the biggest problem was that not every year we could have the yield of the seeds that we would expect. Now the problem is more that those seeds are produced more often, but the quality often drops. So we also have to be aware of that and know about the quality. Collecting seeds for production also is our insurance that the production will be continuously will continue without any delays and problems. Seeds are also, as a product of reproduction, they are a big reserve of genetic variability, which also helps us to create more resilient ecosystem and forest. For more conservation reasons, we know that ex situ conservation, in most situations, not always, but in many cases, is cheaper than in situ conservation, than paying for a big but important arboretums or national parks and botanical gardens. They are space efficient. There is a big variety in sizes, but usually when we think about it, there is a very big genetic variability in relatively small space. And they are, like I said, biologically made to last, because seeds there is this hidden life of them, and the dormancy always helps us to keep them longer safe. The other thing that I like to think about, that you can see here on this picture, is the amount of diversity we are losing. We knew this from the news, from the papers. We hear about this a lot, but maybe sometimes it's hard to imagine, because not all of us can see that there is less herbaceous species, less tree species, that in generally that biodiversity is decreasing. But if we think about, but every one of us sometimes goes to the farm market to buy some vegetables or fruits, and this picture here exactly is an example from the survey that was made in USA, and they check how many varieties of local vegetables disappeared during the 8 years from the market. That's the reason why we have much less different varieties in use or sell in shops. Is very easy. That was the Green Revolution, our great success as a humanity. But also with the hidden cost of that was the reduction of biodiversity. We don't use that many varieties anymore. We have only few that are the best, that has the best yield, they are the most resilient. We don't need that much. Farmers often use only, they don't use seeds from the crop that they produce themselves. They have to buy it each year and each year. That all gets us to the place where we have a relatively cheap food, where we can afford on many different things, but this is a hidden cost, this loss of biodiversity that we can have, that we should keep in mind, and this is one of the reasons that we are also storing the seeds as long as we can. So seeds are usually stored in airtight containers for different periods of time. That always depends also why we are storing them. If we are storing them for some production processes, usually it can be short or medium-term storage. If we store them for conservation, then we would like to store them as long as we can. It is important that we should store good quality seeds, because the better quality of the seeds are at the beginning, the better results we should have at the end. This is always very important. That's why seed screening, seed testing before storage is so important. Usually we store dormant seeds, but here there can be multiple exceptions. Depends on the species, of course, but also there are some techniques where we can store non-dormant seeds without dormancy, that the dormancy was released. But that is usually for a shorter period of time. When we are thinking about the storing seeds, the very important trait that we need to remember is desiccation tolerance. And we can categorize this desiccation tolerance in three distinct groups: orthodox, intermediate, and recalcitrant. I personally like to think about it like a spectrum from orthodox to recalcitrant, because intermediate for me is very difficult to describe and to categorize. It's more, and for you it will be enough if you remember that the orthodox seeds are the seeds that can withstand very strong desiccation, even below 5% without losing its viability. This makes storing them very easy. We just dry them and put them in a very, very low temperature. As they are without too much water, there's nothing bad happening to them during the storage in cold temperatures. And recalcitrant are the seeds that are much more difficult to handle. They remain metabolically active all the time. That's why they need a lot of water. They need to remain moist, very moist, and that makes impossible to store them in sub-zero temperatures, as the formation of ice crystal would completely destroy interior of the seeds and the cells. So in Poland, we don't have many, so we could say lucky we, but those that we have, acorns, for example, are much more, there is a very important timber species which we need to know how to handle. But many species from the tropics, as I know, many that are endangered produce this kind of seeds, and different techniques for storing them has to be applied. But traditionally, or like commonly, if you ever visit a gene bank or seed, or specifically a seed bank, the seed will be stored in a temperature of -18 degrees, and moisture content will vary, depends on the species, but we can say that like safe range would be around 8, maybe even 10%. So all we would like to identify the species that we need to collect, have the seeds, check the seeds. We don't want to store, for example, empty seeds. We want only the best quality, full seeds. We process them, which is usually, if it's an unorthodox species, just drying them to the right moisture content, and then storing in the temperature as long as we can. because the longer we store them, the cheaper the storage is technically. But bear in mind that it depends why we are storing them. If we're gonna use them in the seedling production, or we do it for conservation purposes. And then the recovery, which is with trees is a very tricky part because, as you all know, trees they came into nature very late in our standard, because it's in human standard, because it takes a lot of years. It's not like an annual or perennial plants where the generation comes and go much faster. But even though we have a perfect conditions for storage, it's not like there's nothing happening, that we are like hitting the pause button and then the seeds then just lie in the storage forever, indefinitely. No, seeds are a living organism, and even though we try to slow down the process as much as we can, they still lose the viability slowly. So, and then we go from 100% of germination slowly to much lower numbers. And usually in the storage, we can see the behavior like this. The seeds are okay for extended period of time, and then mostly because of reactive oxygen species, the damage accumulates, and we observe rapid loss of viability, and we need to try to react. Usually, this is the moment that we try to recover our collection. And good condition for storing is the condition in which this first part, when there is no signs of aging, extend as long as possible. And as you could probably figure it out by the classification of seeds, that the two factors that influence the storage the most is moisture content of the seeds and the temperature in which they are kept. This is a nice graph that shows example of how seeds lose their viability in different conditions. This was made on pea seeds, peas, this small green vegetable. And you can see very clearly that as we go with the temperature down and as the moisture content go lower, those curves shows us longer period of storage and allows us to store the seeds safely for longer time. But this is not the case, as I said, with recalcitrant species. A big example that we are using here in Poland and that we are also investigating a lot in our institute is Quercus robur, pedunculoid oak, that you can see here how the moisture content is connected with germination. And this, to make this graph, that was a very nice small experiment. We cut the distal part of the acorn, the one that don't have the embryo, and that way we could check exact moisture content of the seed that we were sowing, and then to check if the seed in the given moisture content was germinating or not. So that we had like no germination, so it was a failure, and germination was a success, depend on the moisture content of each individual acorn. And you could see here very easily why they are recalcitrant and why this moisture content is a matter of death or life for them. As we won't get, half of the seeds will be dead if the moisture content go below 30%. Those two colors indicates two different provenances. They are the same species, the same acorns, but that was Czmoń and that was Siedlce, difficult Polish names. But I lost for some reason, the second name. But as you could see, below 30%, half of the seeds are dead and not usable. But still, if you look closely, the rapid loss of germination of viability goes after below around 36%. And that might be a problem also for us as we observe when we collect seeds in our institute each year, that normally most of the acorns at the moment of the collection was above 40% of moisture content, which was for them very good. And now, very often, even when they fall for trees, they are much, maybe not much, but significantly more drier, around 38, 37. And that also affect their quality and affect their storability. Traditionally, for acorns, the only way that we can store them is short term, not very long, 2, maybe 3 winters, and we do it only for production reasons, to have them some seeds each year to use in the nursery, because this is too short for any longer storage for any conservation reason. Seeds, no, as you could see, the best content for them for storage is 40, 45. We can store lower, but the quality is affected, and the storage temperature is -3 because we don't want any ice crystal formation inside of the seeds. And as they are very metabolically active, they respirate a lot, so the aeration in the containers are always also very, very important. So for many recalcitrant species, including oak, but not exclusively oak, but also different ones, we use technique known as cryopreservation, which is a technique where we put, in our case, some tissues of the seeds to very low temperature, which is liquid nitrogen temperature is almost -200 degrees. This technique is very effective. We have many success with the techniques here in the institute with oak, but also you can find in the literature examples of other species that are conserved like that. But the drawback of this technique is that we need to have a good equipment. We need to have, of course, dewars with liquid nitrogen. Oh, I need to speed up. And of course, to regenerate seed after such a storage, we need in vitro techniques. We can use this technique also for different species. This is example of orthodox species because it can withstand drying, but is very short-lived. In that case, we prefer to use liquid nitrogen than -18 degrees because that's the only way that we can store them for longer. Those are seeds of salix, of different salix species. So, because I need to speed up, we have our storage done. We store the seeds for the time that we want it, and then the next step is to make them germinate. But often, seeds, if we want seeds to germinate, we need to release them from their dormancy. And the dormancy is innate constraint of germination under a condition that would otherwise promote germination in non-dormant seeds. That sounds overly complicated, but it's not. If seeds don't germinate in perfect conditions, it means that they are dormant. And the IMA dormancy is batch heading of the risk of losing too much seeds. We would like to spread germination in time as much as we can, as the seedlings are very fragile, so choosing for the seeds, for the seedling, choosing the right moment to germinate is this is a matter of their survival. What is important when you work with seeds, that to keep in mind that even seeds from the same plant can have a different level of dormancy. Some plants, some species have very polymorphic seeds with different levels, and that makes the germination then in practice could take extended period of time. Here is few information about types of dormancy. I will maybe not dive into too much details, but dormancy types are important to know. We should study them for each species because this is the only way that we could mimic the condition for release the dormancy and use them in practice to have as much germinated seeds in nursery as possible. Such experiments were made also in our institute, mostly, almost exclusively for our native species from Poland, and then depending on the type of dormancy, if it was normal physiological dormancy, we adjust the temperature and the time that temperature is affecting seed to receive germination. For example, at least one example, the very interesting situation is Fraxinus excelsior, which has morphophysiological dormancy. Seeds, when they are dispersed in the nature, even though they are mature, the embryo inside the seed is not mature. It needs 16, depends, but let's say around 16 weeks of warm temperature. We even try to calculate the optimum temperatures, which the optimum temperature is around 16 degrees, for them to grow, fill up the seed completely, and then still we need another 16 weeks of cold to release the physiological part of the dormancy. So, okay, our seeds were stored, they are viable, then dormancy is released. So the last part is germination. And germination is tricky because germination responds to the same environmental cues as dormancy, but it's a completely opposite process. Yeah, the dormancy tends to keep seeds alive, but not germinating, and germinating is the moment where the seedling is appearing. But the biggest difference between dormancy and germination is that the germination is an irreversible process, cannot be stopped. We can like slow it down, but we cannot stop, we cannot go back after this. And germination usually have 3 phases. 2 first phases are not visible, all is happening inside the seeds, in the cells, and then only the last part is the moment that we can see the embryo going out, the root start to growing. It is important to know that those 2 phases are invisible, but make the cells inside the seeds active, because this is the moment that we can, for example, have some additional pretreatment, for example, priming of the seeds, which also can later even and synchronize the germination in the nursery. So this is something that I said, that the dormancy and germination respond to the same environmental cues, but they are opposite processes. Germination is irreversible and dormancy can go in the cycle. Seeds can induce the secondary dormancy and release it depending on the environmental condition that they are in. And this is the last almost. Because this is a very nice metaphor how we can think how the seeds receive the signals. It doesn't have to apply only to seeds. We can think about buds and other stuff as well, but of course for this presentation we will focus on the seeds. It was prepared by Bradford in 2000s, and this is a very nice model and very nice paper. But anyway, if we imagine that this big tank is a seed population and there is some environmental signal like temperature, the seeds are accumulating the signal to the moment where they have their minimum amount of the signal that force some response. This response in that case is germination. So if we accumulate enough signal, the germination starts. And all the seeds start to germinate. This is this is one model, but in reality, always the seeds are much more complicated. They are more polymorphic. So this is more than only one threshold that make them germinate. If we have the same signal, part of the seeds in the population will start germinating earlier, then another part later, and so on. This makes them more resilient to the changes, because if the signal will stop for some reason, we will have a very cold or very dry spring. Not all seeds will germinate. Part of them will stay in the soil, and who knows what's going to happen with them. They can be eaten, they can be dead. A lot of things can happen with them. The question is if they can adapt, if their responses can be reached in the lower signal. That we don't know. That is something that we still need to evaluate and study, especially in trees, as they are much more, they're long-lived organisms. This I can skip and finish the last thing. I know everyone is hungry. Me as well.
Mikołaj, red card in last minute hurts very much. So,
a part of the things that I said, very important is testing the seeds, because if we test them properly, we know what we are working with. But to have a proper tests, we need to have a good knowledge of those of seeds. Without knowledge about seed physiology, we won't have a good testing. We won't have proper seed handling, which will result in losses and waste. And my take-home message would be like better seed knowledge, we would have better forest. Thank you very much. And sorry for the delay. Sorry so much. Yeah.
Thanks to Mikołaj, we are almost right on time. So thank you all for being the part of this session. Thanks for our speakers for almost perfect time organization. And we have one announcement for you.
Even two. The first announcement is about the lunch. So the lunch will be served like soup will be given to you when you will be sitting near the table. So please take a seat, and the main dish you will be able to take by yourself, choose whatever you want. And the second information is for all of you who stays here in the hotel, because the keys to your room are available. So you can go to the hotel receptions and take your keys. Okay, so it's all. So we can start next session after the lunch. I hope soon more people will come to join us. In this session we have 3 lectures. The first one will be given by Professor Teresa Chazubska-Przybył. She will take you to the world of the in vitro cultures, the world which is covered with forests of tomorrow, modern forests. So the floor is yours.
Thank you. Good morning, everyone. I would like to invite you very kindly on this on my lecture, and I would like to present my results of the studies on the somatic embryogenesis and cryopreservation in conifer species, which I am carrying on in our institute from 2000 years. And I would like to show you that this both methods are the method which integrates biotechnological advances with practical forestry. Let's begin with this picture. We know that today the forest is under a great pressure. It is a great problem in all the world because we can see such pictures like you can see here. For example, in Brazil, in the tropical forestry, also in Europe, like in Italy, also in Poland, like in Białowieża National Park, where our Pisa abies is dying due to the climate change, and also even in our arboretum in Kórnik, when we can see that the spruce which I'm working on from when I started my work, I started work with Pisa omorica, and it was always in good condition, but now we can see that even this species started died. We think that we can have one of the solution to resolve these problems using, for example, biotechnology tools like tree micropropagation. It is a very attractive method because it enables a rapid clonal propagation of selected genotypes, select the plants which are uniform and which are disease-free, and we can use the in vitro culture for this purpose, for example, somatic embryogenesis and organogenesis, and in this way support the conservation of the allied genotypes and also support the forestry. In vitro, this method was started at the beginning of the 20th century through the Austrian botanist Haberland, who introduced the idea of plants which will grow in the artificial environment, and he called this idea the theory of totipotency. What is the totipotency? It is the idea that plant cells can have the possibility to divide and to produce the specific organs and also the entire plant organism. And based on this theory, also the somatic embryogenesis as one of the vegetative micropropagation method was developed, and it is especially useful for cloning allied trees. And what is it, the somatic embryogenesis? It is the regeneration of the plants through the somatic embryos from which are induced from vegetative cells. It is a very complex method because it includes a few phases. It is the first phase. It's the induction of this process from an explant taken from the donor tree, which in conifers especially are immature or mature zygotic embryos. In the next phase, the embryogenic tissue is initiated, and next the tissue is proliferated under in vitro condition, and next it is placed on the special media to regenerate, to induce the maturation of somatic embryos, and in the next step to induce the germination on the other condition, conversion in the somatic seedlings, and finally to obtain acclimatized somatic seedlings to the ex vitro conditions. Another biotechnological tool is cryopreservation, which is also useful in the combination with somatic embryogenesis in conservation of valuable genotype of trees. And as Mikołaj said, it is a storage of the plant material in the sub-zero temperatures, like, for example, minus 19.6 degree. And in this condition, plants may be stored for an unlimited period of time. And this is the way when we minimize the risk of the loss of genetic stability and also the contamination of the plant material. And this method is used for both for the broadleaves and for conifers tree species, mainly to store the plant material in form of the somatic embryos and embryogenic cultures. The combination of somatic embryogenesis and cryopreservation enable us to save and at the same time to test the obtained somatic plants in the field tests. Embryogenic tissue is a special plant tissue which contain immature somatic embryos. Here we can see the special structure of these embryos, which are able to survive the cryopreservation procedure, and after the cryopreservation step, we can also obtain the somatic embryogenesis after the thawing of the embryogenic tissue from liquid nitrogen. And in the next step, we can also produce the somatic embryos, which can be used for the next phases of the somatic embryogenesis to obtain the implants. In Poland, and especially in our institute, we studied mainly Pisa abies and Pinus sylvestris as the most important species for the forestry in our country, and also the Pisa omorica, in comparison to the PISA ABIES, which is the model for the studies on the somatic embryogenesis all over the world. And PISA omorica is also very interesting for us because it is an ornamental tree, and it is also endemic for the Serbia, Croatia, Herzegovina. So it is also very important from the ecological point of view. But the induction of the somatic embryogenesis is not so easy. We tried do this using the mature zygotic embryos taken from the seeds. And you can see here that using the different media, we can obtain only maximally about 20% of the explants with embryogenic tissue. So it is only 1/5 of the explants placed on the induction medium. And you can see here also the immature somatic embryos, which has a special structure. There is the embryogenic region. When we stain this tissue with the acetocarmine, we can see the isodiametric meristematic cells from which the somatic embryos will develop in the future, and also the longer, highly vacuolated suspensor cells, which transport the nutrients from the media to this developing somatic embryo. In the next step, also, it is the need to improve, to optimize the maturation protocol. So we tested different concentration of abscisic acid, which is necessary to induce the somatic embryos germination from this immature embryos, which are visible in the embryogenic tissue. And we also have to support the development of the embryos using the mild osmotic stress, for example, through application of the sucrose in different concentration to the media. It also depends on the species and even the cell line. And here we can see that the germination percent was high both in PISA ABIES and PISA omorica, but the next step, conversion of this germinating somatic embryo into plant, was very low. It was only 2 and 3%. So this step is also very problematic to obtain the full developed somatic seedlings. And during this process, there are different physical factors which regulate somatic embryogenesis process in conifers. On the initiation is mainly the temperature. During the proliferation, the oxygen gases conditions. During the maturation, the humidity. And next, during the germination, the light, and also the osmotic stress. And when we obtained the germinating somatic embryos, we can transfer them from the in vitro culture to the ex vitro conditions, and we use to this aim the special jiffy pots, which are filled with the special soil dedicated to the conifers plants, and in this way we can obtain in a few months the growth of the somatic seedlings with the well-developed needles and the buds, which are able to develop into in the next season in the well-developed imblings. And finally, we can obtain also the full developed plant, somatic plant, which can be transported to the field in the forestry for testing how it is their response to this natural environment. During the somatic embryogenesis process, we have to also monitor the quality of each of the plant material at each stage of the somatic embryogenesis. For example, when we tested the influence of the ascorbic acid on the proliferation, we have to check also the oxidative stress level, also the hormones levels, and antioxidants which appeared during the particular stage of the somatic embryo development. Also, after the cryopreservation, we have to develop, we have to control the genetic stability of the plant material. Here you can see we did it, for example, for the five I do not remember. Never mind. For the 5 microsatellites, yes. And you can see here that in this plant material we tested in this studies published in this article, we didn't find any changes in these places in the DNA. So it suggested that we obtained the unchanged plant material as a result of the cryopreservation we used in our laboratory. And this is the question, does the cryopreservation really work? I had a possibility to see the special cryopreservation place in the Luk Institute in the Finland when they have a special equipment and special rooms for this aim, and they could storage in the liquid nitrogen thousands cell lines obtained from the somatic embryogenesis. So this is a very, very important and very useful tool in saving also the plant material obtained under in vitro culture conditions. And here you can see also that somatic seedlings may be maybe cultured also in the forest nursery. This is also in the Look Institute. You can see the 2 and 3 years old somatic seedlings of Serbian larch and Norway spruce, which are prepared for the transport to the forestry area, and also the experimental plot of somatic trees of Pisa abies, which were monitored each year in this place by researchers. And it is always, we have to aware that we have the great challenges using the biotechnological tools in the micropropagation, especially due to the biological limitations, because each step of somatic embryogenesis and also the cryopreservation is under the strange genotype dependence. And also the induction of the process is limited because we have the possibility to collect the explants only seasonally, and they have been in the special psychological phase. And also it is another problem and another limitation is the complex genomes, which have coniferous species like Pisa abies and Larix sibilca, so it is not so easy to optimize the protocols of in vitro micropropagation. And another limitations are of technical nature. It is a law. One of such difficulties is low somatic embryos efficiency. Also the problems with the surviving of the cell lines during the cryopreservation, and overall the system complexity, because it is difficult to optimize it and scaling it up. And future directions should be the protocol optimization and also advanced monitoring at the biochemical and molecular levels, improved cryopreservation efficiency, and also the transfer plants from the lab to the forestry field. So, in the broader perspective, in the end, this presentation is not about the trees, but about the ecosystem resilience and biodiversity, and also our ability to respond to the environmental change. And this message, you should remember that the somatic embryogenesis and cryopreservation are very important tools that enable the propagation, preservation, and protection of the forest genetics resources, and they are important also for the future of sustainable forestry. Thank you for your attention. And that's all.
Thank you, Teresa, for this lecture. The next talk will be given by Weronika Żukowska. She will tell us about the conservation of the genetic resources of forest trees. And the title is a little mystery because she will focus on 2 species. And up to now, we know that they are only contrasting species. So let's find out what the species are.
How's your blood sugar level? Yeah, manageable. Okay, so my presentation will be about conservation genomics, but instead of giving you the principles or good practices, I decided to talk about 2 different species and give you a little bit of a story just to, you know, move you a little bit after the lunch. So, starting with the pyramid of biodiversity, I think you know it. There are 3 levels of biodiversity. And the very basic one is the genetic diversity, which is the foundation of biodiversity. Then we have the species diversity level and the ecosystem level. When we talk about trees, they need to face numerous challenges right now, such as climate change or habitat fragmentation, and of course, human pressure. But when we think about the genetic diversity of trees, we need to consider the fact that they live very long, their generation times are very long. So for trees, for the genetic changes to appear, we need to wait a lot of time, which we don't really have right now. But there is also another but to this story, is that what we want to ultimately preserve are not the genetic resources themselves. We think about resilience, which was already mentioned here. And the resilience is not only a matter of genetic diversity. There are different processes that need to be guaranteed in order to have the ecosystem resilient. And I named just 5 of them, which you can read by yourselves. And only if they work together, the forests stay resilient, and this means that they can recover after some kind of a disturbance. The main factor that form the genetic diversity of trees are these 5. The 2 basic are mutations and recombination and sexual reproduction. The 3rd factor is gene flow, which is probably the most important for forestry species. And gene flow is here understood by the movement of pollen and seeds. And then we have the factor connected with some random changes, which are called genetic drift, and those which are connected with the adaptation, so this is natural selection. And as I already mentioned, the genetic diversity of trees is mainly shaped by the gene flow, and typically we have low genetic structure between trees populations and very high genetic diversity within the populations. But what we see today, I mean the genetic diversity that we have today is not only the thing of the current things, is also the historical legacy and many different factors that happened also in the past. So we can name a few of them. Of course, this would be glacial cycles and post-glacial recolonization. Then we have gene flow, which happened in the past and which is happening right now, and some demographic history of each population, which in each case is different. And then, of course, we have some anthropogenic drivers of genetic diversity, among which we can name habitat fragmentation, some management practices, movement of planting material, overexploitation, or the pollution. And for each species, of course, this factor will differ, and that's why we need tailored approach for each species. So let's meet the 2 main characters of my story today. This will be Scots pine and black poplar. What they have in common is that both of them have broad distribution, mostly throughout Europe and Asia. But when it comes to Scots pine, it forms large continuous populations, whereas black poplar is a specialist which is connected to river habitats. Scots pine has very high genetic diversity and phenotypic plasticity because it occurs in various different environments. And when it comes to black poplar, it still has high genetic diversity, but we see more and more clones in its populations because it can also reproduce vegetatively. Okay, so the power of Scots pine is its connectivity. And at first glance, what we see are stable forests, which are relatively secured. But when we take a closer look, we may find some distinctive populations, isolated ones or marginal, typically located in mountainous regions, and they usually have some unique adaptations, which are very important for the whole species. And they are usually isolated, so the gene flow is weak, and we have reduced connectivity. And here are the results of one of our studies when we assessed the genetic variation of, I think, 60 populations of Scots pine. And here you have on these panels the expected high heterozygosity and allelic richness of these populations. And as you may see, for most populations it's pretty high, but there are places when it's lower, especially in Italy and in Romania, in these mountainous regions, or in the eastern part of the range, which is probably the effect of the founder effect. When it comes to genetic structure, the most probable number of genetic clusters are 2, which represent European and Asian populations, but here we have some subtle substructure, 5 genetic clusters, and if you take a closer look, we have some unique genetic pools which correspond to Spanish, Turkish, and Italian populations, which are located. This is the result of the mitotypes, so the mitochondrial DNA, which is spread by seeds. And here we revealed that we know that glacial refugia for most species were located in the south, but there are some cryptic refugia also at higher latitudes, as you may see here, for example, in the south of Finland. Okay, so now moving to black poplar, what appears to be a large population and a healthy one, may in fact consist of clones and hybrids, which are usually impossible to detect just by morphological features. So what we need are dynamic rivers in order for the seedlings to appear, because they need some specific environmental conditions to germinate. And right now we don't really have that huge floods. And what's happening is that clonality starts to dominate, and we have more and more hybrid pressure. This is the result of our study, which was conducted in Poland along the 6th biggest rivers. This is also about the genetic variation of populations. And as you may see here, it's generally lower in these lower parts of the rivers, where we have higher anthropogenic pressure. Also, when it comes to clonality, we found that it's correlated with the level of river transformation. It's higher, especially in the Oder and Varta, upper and lower Vistula, and these parts of the rivers are highly regulated. The least regulated is the middle Vistula, and Sandbuk and Narev also are less regulated, and you can clearly see here that clonality is lower. When it comes to genetic structure of black poplar, you can see an obvious genetic structure along Varta, Oder, and Buk. There are some unique gene pools, as for example number 12, number 25, I believe. And relatively free gene flow appears to happen only along the Vistula, which is probably the last place where we still have natural generation, which is generative. So we're not really happy about that, and we need to monitor the situation each year. This is also the genetic structure, but with different kind of markers. These are SNP markers, which are supposed to show more information connected with adaptation. Here on the left, you see all populations, so they cluster pretty much the same. We have the Oder catchment and the Vistula catchment, but there are some outliers, especially along the Varta. and you have it on the right. Yeah. And from this, you can see that there is something distinctive in here, and when we check this population carefully, it's probably an artificial population which was planted by human. There were only a few genotypes there, and these were not genotypes which are typical for the western part of Poland. Whereas in here, probably this is a signal of some kind of an adaptation, but we are still analyzing these results, so we're gonna see. When it comes to hybridization, it feels like the risk is pretty low. This is one of the examples of our tests. We tested here more than 200 seedlings from the Middle Vistula, and we detected only 3 probable hybrids, but we should keep in mind that it may change in the coming years because hybridization risk is connected with environmental conditions. So when they change, the phenology may change, and we can have more hybrids in the coming years, or less. So what are the management implications when we think about the 2 species? We have the same goal. We want to preserve their genetic resources and ultimately their resilience, but we need to have different pathways to do that. For Scots pine, it would be very important to maintain connectivity with those isolated populations, to identify them and protect their gene pools, and this is quite challenging. One factor is that we need to sample these populations. These are usually difficult to access, and the other one is that those adaptation signals are really hard to detect, but anyway, we need to try to do that. When we want to do things like reforestation, we should use as much diverse seed sources as we can, and in some cases it might be wise to consider assisted gene flow. When it comes to black poplar, it would be ideal to restore river dynamics, which is generally impossible. So what we can do is to try to promote generative regeneration in the most appropriate places and do some kind of a genetic monitoring to detect clones and hybrids. And for this species, it would be wise to consider ex situ protection, such as clone archives. So here I got to the key message. The resilience is not determined by genes alone. We need to think about other processes that interconnect. And even widespread species can be vulnerable, but in different ways. Thank you.
Thank you very much for keeping the time. And I would like to invite Francesco Latrini to give the lecture. He will tell you how to work smarter and not harder in the forest management. And he will outline that it is possible by using the modern and available tools.
Levo. Okay.
Okay, first of all, I would like to start with a good news. I am the last keynote speaker, so it's almost done. Please, hold on, in some minutes we'll be finished, and try not to fall asleep. This is the starting point. As Eva told, today I will be speaking about how remote sensing can help us in shaping smarter and more sustainable forest operations. First of all, what I mean with forest operations? With this term, I mean the practical way in which we are carrying out the forestry. So, which kind of machinery are we using, where this machine can or cannot go, and in which way. Sorry. First of all, I would like to introduce the technology that I will be speaking today. So, LiDAR, which is the acronym of Light Detection and Ranging. It's very similar to the much more known radar that every one of you knows from, I don't know, war films or flight disaster movies, etc. is a technology which is using laser pulse emitted by a sensor. This sensor calculates the return time from the moment in which this laser pulse hit an object, in our case, a tree, the tree crown or the soil. Calculating the return time, it creates the so-called LiDAR point cloud, which is a model, a 3D reconstruction of the forest and of the soil. I will tell that LiDAR is the most, the most powerful remote sensing technology that we have now in forestry. Why? Because it's the only technology now which is able to penetrate the canopy. So we, at this moment, even from a certain distance, and later I will explain what I mean with certain distance, we cannot even have information about the canopy layer, like we can do with many different satellites, for example, but we can even get information about the soil. In these images, you can see a point cloud, a LiDAR point cloud acquired by a drone. On the left side, the aerial view of a beech forest. On the right side, you have the section of the point cloud after the classification. What is a classification? Is the application of some algorithms which allows us to understand which of the millions of the points which belong to the cloud, which belongs to the soil in green, or to whatever else, in our case trees, in blue. Thanks to this point cloud, we can use some interpolation algorithms to obtain 3 fundamental layers for planning the forest operations. These are the DTM, Digital Terrain Model, DSM, Digital Surface Model, and the difference between the two, Canopy Height Model. I imagine that the first 2 pictures on the left are not very informative, but I can believe me that in the first one, each pixel of the raster contains an information about the elevation of the soil above the sea level. The second one contains the same information, but of the upper surface, so in a forest, the trees. The difference between the two allows us to obtain the canopy height model that, as you can see, is much more informative. There are a lot of trees. This is a beech forest in central Italy. And each pixel contains an information of 3 height. LiDAR in forest operations can be used via 4 different platforms. There is also now the space-borne LiDAR from satellite, but it is too coarse to be applied for forest operations. It is more about biomass estimation on a large scale. For our goal, we have to refer to airborne laser scanning, so a LiDAR sensor mounted on an airplane or helicopter, UAV laser scanning, which is LiDAR sensor or a drone, and here we are in the so-called remote sensing, so we are hundreds of meters above the canopy with ALS and dozens of meters above the canopy with the drone. Mobile laser scanning and terrestrial laser scanning are the so-called proximal sensing. So we are much closer, we are, however, inside the forest. What changed about moving, let's say, from left to right? We have a strong increase in the resolution of the data that we can obtain. Just to give some number, a digital terrain model obtained by airborne laser scanning has a pixel of about 1 meter of resolution. Arriving to the terrestrial laser scanning, we can arrive to about 1 centimeter. Obviously, the surface that we can cover with one survey moves oppositely. So if with the airplane we can cover thousands of kilometers in one survey, with the terrestrial laser scanning, more or less we are like a conventional ground-based survey done with the conventional technologies. After this introduction, I would like to tell you that this LiDAR technology can be used both before the harvesting for planning the interventions in a smarter, in a more precise, more sustainable way, and also after forest operations to evaluate the possible environmental impacts that are, unavoidable when we are dealing with a machine driving in the forest. What can we do before the entrance of the tractor or whatever inside the forest? We can, thanks to the digital terrain model, identify with a very high precision, with accuracy reaching 99%, the previous skid trails. So we can know exactly where 10, 20, even 30 years ago the previous machinery went, and so we can reutilize directly these skid trails in order not to create further disturbance to the soil. If these skid trails are not enough, we can plan a further trail network, however, starting and taking into consideration what already is. Even more, now there is a powerful tool developed in the Nordic countries, which is called soil trafficability mapping, which is a further raster layer developed from the digital terrain model, which estimates somehow where a machine can be driven into the soil without creating excessive impacts. I will not go very much in details about this because in the next days we will see some presentations about this technology. Finally, if we have the digital terrain model, we can have information, as I told, about where the trails are, where the soil can be trafficked, which is the slope of our terrain, which is the roughness, so the percentage of soil which cannot be afforded by machinery. And in this way, we can plan from our house, let's say. Still, we can somehow plan what can be the best harvesting system, the best machinery to be used for the intervention. This is a nice example that I would like to show you. This is a possible workflow LiDAR-based for forest operations planning, referring in this case to uneven age forest management, so single selection and group selection cutting. In red, you have the theoretical location of the trees to be extracted, or groups to be extracted, that I can derive from the canopy height models, because as we know, in the single selection cutting, the trees that have to be subject of the operation are the biggest one, so most probably the tallest one, and so I can extract the position of these trees from the canopy height model. From the digital terrain model, so the one of the soil, I can identify the previous trails in purple, the areas which should not be trafficked by the machine. You can see these trafficability maps in this shading blue. And so, knowing this information and knowing the machine that I would like to use in this kind of situation, I can develop the green trails, which are tree or group-based trails for the extraction of the wooden material from the parcel, which allows to minimize the impacted surface, to not traffic the machine on excessive slope, so also to taking into account the safety for the operator, for the driver, and also minimizing as much as possible the soil disturbance. As I told you before, however, we can even do a step further and use this technology after the forest operations. So if we do a further survey after the end of the silvicultural treatment, and we obtain a new digital terrain model, digital surface model, and canopy height model, we can, apart from obviously identifying the cutting, so where the trees were removed, if we are interested in this. But what is very interesting, considering that the biggest impact related to forest operations on the forest ecosystem is the soil impacts, so soil compaction, rutting, soil displacement, etc., we can run again the algorithms that we ran previously to identify the existing trails, to identify the new established trails. Are these how to say, well positioned? Are these too many? Are these too deep, etc., etc.? And this still from ALS or drone-based LiDAR. In the case that we have also the terrestrial asset scanning, or even better, the mobile asset scanning, we can go much closer and really do a quantitative evaluation of the depth of the rutting, as you can see in this photo. So you can expand somehow the evaluation of soil compaction and soil rutting, which generally is punctual, all along the trail network. And so now the questions. So if everything is so beautiful, if everything is so functional, why, if you go to speak to the major part of practitioners, apart from the Nordic countries, why it's not used? Because what I show till now is it's everything ready. This is not anymore research. We are in the phase of, how to say, of I will not tell commercialization, but exploitation at the practical level. Is the moment that scientists should train somehow, should explain to practitioners how to use these technologies in order to have them applied every day. However, still there is, in my opinion, a little bottleneck before this. And this is related to the fact that some of these applications that I'm showing you, most of all the one which does not rely on the raster layers, but directly on the point cloud, requires some very powerful hardware. So I'm working with this data every day. I have a dedicated computer which is done and thought to manage this kind of data. Without this, with a, how to say, custom hardware, it can be difficult to work on the point clouds. But this does not apply for the raster. So the ATM, DSM, etc., etc. And the other, how to say, bottleneck, in my opinion, is that many applications are still based on coding via R, via Python, etc. So the next step to make this really applicable should be to collaborate also with other sectors like informaticians and develop dedicated softwares or even plug-in for existing softwares which make it easier and faster the application for the practitioners. And I think that, however, soon we will arrive to this. The last suggestion that I have, but this is even at the policy level, is to enlarge the cover of the national ALS programs. For example, Poland, I like very much to work in Poland because there is the ALS survey very updated every 2, 3, maximum 4 years for all the national surveys. Not in every country is like that. So this also, the availability of open access data will strongly, how to say, foster the applicability of this technology. If you want to go deeper, there is this review that I published previous year with 2 colleagues from New Zealand, in which all these technologies are discussed much deeper and in much bigger details. So I invite you to download the paper, it's open access, download, read, and if possible, cite. And nothing. Thank you very much.
Thank you for this interesting talk. And right now we have another coffee break. But I have the one information because each participant of this conference is invited to the tour to castle. And just after the dinner, everybody who wants to go there should be 15 minutes after 8 before the main entrance to the hotel, because you will go there on foot. So wear something comfortable, because you will go on the promenade along the lake and the walk will be around 30 minutes. So be ready after the dinner to go there. And right now we need 20 minutes to rebuild this space into 2 separate rooms for another session. But I'm sure that it will be something sweet given during the break, a dessert, not only a coffee.
Raz, dwa, trzy. Raz, dwa, trzy.
No tak, z tego co wiem, to tak.
I gotowe. Ja ją wyłączę, nie? Bo to nie trzeba się bawić, że w tym systemie.
Nie ma, bo oni kręcą.
Raz, dwa, trzy. Raz, ale jo. Raz, dwa, trzy. 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 Raz. Raz, dwa, trzy. 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 Mamy troszkę słychać, musimy cicho robić. Minimalnie się... On to ciszy. I tutaj też ciszy. Raz. Na sali musi być ciszy, bo my tam słyszymy. I ja tak samo. A już słyszałeś ten stream? Nie, w streamie nic, nie? Bo ja muszę kliknąć play, nie? Ale muszę gdzieś do ciebie coś? Nie wiem. Raz, dwa. Gdzieś słychać. Raz, dwa, trzy. Ja tam będę widział, nie? Tomek, wchodź, więc jest wszystko widne. Tylko jedna rzecz, trochę ciszej. Kadrowanie tak naprawdę na detal, nie? Nie, tam coś widziałem, nie? Ja, no.
All right, so I welcome you on the session. We are in room A, so I hope that everybody who wanted to be here are actually here. And we are started with the quite interesting species, Fagus sylvatica. So I invite Andrzej Nezval, I hope that it's good pronunciation, who tell us more about temperature-driven onset and light quality linked senescence in Fagus sylvatica phenology. Okay, so we will switch. And here you have your presentation. And you want to have it in hand or here?
It's okay. Hello. Hello. I wish you good afternoon. My name is Ondřej Nezval, and let me introduce our research about phenology observations. Let's start with introduction. Why I study phenology? Because we are facing climatic changes. Across the Czech Republic, we can see significant shift in temperatures, rapidly increasing air temperatures, and shift toward warmer climatic regions, as you can see on this picture. Why phenology? Phenology is a good tool and indicator of climate change. As you can see on this part of the slide, you can see temperature anomaly across Europe since 2018 to 2025. And you can see how beech forests reflect actual climatic conditions and climatic variability across years. And on the end of April and the same day during years. So forest well respond sensitively to climate variability. And you can observe phenological shift which provide a direct window to ecosystem responses to climate change. There are some open questions which we are tested, and some research and aim of studies to identify ecophysiological thresholds controlling beech growth, quantify time lags among phenological and physiological processes, and link these thresholds to local climatic conditions. There is some description of our ecosystem station, Štítná Plot. It's on the southeast part of the Czech Republic, in White Carpathians in the Czech Republic, as I mentioned. It's dominated by mature European beech, Fagus sylvatica. Standard age is around 100 years, and elevation is around 500 meters above sea level. Annual air temperature around 7 degrees Celsius, and precipitations around 800 millimeters per year. We connected in our research climate conditions, phenology research, and physiology framework, respectively physiological measurements by edicovariance, dendrometers as secondary growth, and phylogenesis as a cambial activity, start of cambial activity, cell enlargement, end of secondary wall thickening, and phloem collapse. And we observed classical ground-based phenological stages like bud break, to fully develop leaf area, to leaf coloring, and leaf fall, as you can see on this picture. So we connected classical phenological with climatic drivers and with physiological responses. We compared long-term data according to interpolation data from nearby Czech Hydrometeorological Institute stations, and you can see significant increasing of air temperature across decades in case of the Czech Republic. It's alarming, more than 2 degrees Celsius per very shortly period, as you can see here. In 1960s, there was a temperature around 6.6 degrees Celsius. In the last decade, you can observe more than 8, respectively 9 degrees of Celsius in this elevator. And we are observing a significant shift in temperature and more unusual warm days across years. And you can see shift temperature. We compared long-term data, as I mentioned, since 1960, and we observed a lot of alarming significant shifts. For example, decreasing ice days, its mean Tmax below 0, frost days, Tmean below 0, and significant increasing tropical days and summer days. It's represent a maximum temperature about 25, respectively 30 degrees of Celsius across years. There are some patterns. If we compared long-term data and last decade, you observed that there are some drought periods, especially on the spring, on the middle of summer, and on the first part of winter, and very significant increasing air temperature during winter and summer period. And a very intensive seasonal drought, as you can observe this year across Central Europe. There are some meteorological data, as you can see on this slide, and you can see significant shift air temperature, unusual warm yearly temperature across our observations during 2018 to 2022, temperature anomaly and annual precipitation according to long-term period. Only 2020 and 2021 was relatively normal, but still unusually warm, as you can see on this picture. Yes, of course, another problem is shift, respectively prolongation growing season. We observing this problem on ecosystem Štítná, our research plot, and you can see significant prolongation of growing season across our observed period according to long-term period. It's alarming that warmest part, growing season, it's mean representing dates with average daily temperature about 15 degrees Celsius more than month. More than month. So you can see enormous prolongation this warm part of the growing season. And general trend in the Czech Republic, you can see on this picture, prolongation around 1 day per decade since 1960. There are some general results, as you can see, and as I mentioned previously, we observed classical ground phenological stations like stages like bud break to leaf area development, to leaf coloring and leaf fall. And we compared these thresholds with temperature-based growing season and seasonal trajectory, which we monitored by phenocams. And phenocam thresholds, phylogenesis, as I mentioned, start of cambial activity well reflected bud break, and you can see best matches, respectively best thresholds, and bud break will represent start of cambial activity, and foliage formation, leaf area development represent start of GPP and subflow onset. And very interesting thing, we found reaction beech forest on light during late summer, and days around 2010, respectively 2050, will reflect respectively clearness index will reflect phloem collapse and end of cambial activity, and clearness index will reflect starting of leaf coloring, as I will show later. And there are some different thresholds which well correlated with phenophasis stages, like 10 degrees Celsius threshold for this research plot. This is a very interesting thing, like summer light started leaf coloring. We observed then during these days and 7 window, 10 window and 7 respectively 14 windows average of clearness index. It's represent something like cloudiness above the sky, and it's represented that more respectively highest clearness index shifted autumn phenological stages. So it's mean that beech forest well reacted on a light on the end of summer. So we can see very interesting correlations. Start of leaf coloring 10% on the axis Y, and clearness index on the axis X, which represent cloudiness during this period. So something like conclusion, key findings which we observing extreme weather conditions we observed across years 2018 to 2022. If we compare with long-term period, more than 2 degrees of Celsius increasing annual temperature and the degrees of rainfall precipitations, prolongation growing season in extremely years, more than month, hottest part of the growing season. We confirm that phenology is a good climate tool, and temperature threshold 10 degrees Celsius well reflect bud break cambial activity, start of GPP around Joyce 112. It's around middle of April, and another results we can discuss personally. So after my presentation. Thank you for attention and have a nice rest day.
Okay, thank you very much for your talk. We will have time for discussion at the end of this session. So now we are skipping and focusing on another species. So I invite Shirin Alipur, who tell us more about Iranian oak habitats and space. Please.
Good afternoon. My name is Shirin Alipur. Today I'm presenting the Iranian oak habitats and space, a combined gap analysis to inform the effective protected area expansion. In Iran, the majority of the oak species is across the Zagros Mountain, Hyrkanian, and Arasbaran. In this study, we focus on the two important species. First, it's Quercus branti, which, as you can see in the map, it's distributed across the Zagros Mountain. The Zagros is the largest forest area in Iran, which is dominated by this species. But I have to mention that Persian oak, besides the Iran, also occupied in Iraq and also part of the Turkey. The characteristics of this species is high tolerance and elevation range up to 2,700 meters. The second species is a Caucasian oak or Quercus macrantera. This species is native in Caucasus and southwest of Asia. In Iran, it's occupying the Hyrcanian forest and Arasbaran, which these two regions are internationally recorded because of their biodiversity and ecological importance. The Caucasian oak, the most important characteristics is tolerance to cold and semi-arid condition, and also the elevation range is quite high, between 1,500 to 2,000, almost 900 meter. As everybody knows, right now, the two most important factors, climate change and habitat fragmentation, threaten biodiversity. But the interaction of these two characters for impact of these two characters on some species like oak, specifically in the Iran, is still poorly understood. And in Iran, most of the study right now is mostly focusing on the future projection. And besides, however, the connection between the historical climate change and the protection area still underestimated. According to these 2 points, we can say that two questions is overlooked. One is that how past climate could shape the current oak distribution and refugee area, and this answer to this question could help us to identify the refugee, which could reveal the hotspot which is important for the genetic diversity and long-term survival. And the next point is that are those protected areas, which in Iran is already administrative protected area, is sufficient to conserve the suitable habitats. But according to 2 points, that in Iran currently almost 10% of a protected area defined, but still many unprotected unsuitable habitat is not included in this defined protection. And the second point is that the Department of Iran plans to expand this protection area. That's why the systematic gap analysis could be required in this case. In this study, we try to answer these two questions. For this purpose, we follow this method. First, we collected the occurrence data for both species. Then, for modeling the distribution, we use the maxent and we project this species distribution for a past, current, and future. For landscape fragmentation and connectivity, we use the toolbox. For priority area, we use the zonation, but for the assessment of the protection overlap between protection area and high suitability habitats, we use the Venn diagram, which allow us to evaluate the effectiveness of existing protected area and identify the gaps that where conservation coverage is insufficient. The results showed that the precipitation is one of the most important environmental factor to shaping the range of the both Quercus species. And as you can see, the result of the current condition, it showed that the Quercus branti covered the highest suitability in the north of the Iraq and all the Zagros range. But for the Quercus macrantera, it showed the border distribution, which not only it's occupied whole the Caucasus, Hyrcanian region, and plus the Arasbaran. But for the future projection for both species, project the loss of the habitat range. For Quercus macrantera, up to 30% of the high suitability range may lost. But this is parallel with a moderate air shift of this species to higher elevation. For Quercus macrantera, as you can see, this species, it might be the greater risk of the lost, the stable habitat range, and so it means it showed that this species is more sensitive to climate change, and as you can see, it's projected the stable range will remaining mostly in Caucasus and Hyrcanian forest. Through the LGM, the fragmentation for both species happened. For Quercus macrantera, unsuitable condition along the border of the Iraq and Iran, the discontinuity range for Quercus happened, and it make the west and eastern part, and it looked like that, making isolated population mostly in the Zagros range. For Quercus macrantera, it showed the maintained the border distribution and persist in likely in the refugia area, which those hotspots it's in Hyrcanian forest, Caucasus, and parts of the Arasbaran region. The analysis of the fragmentation and connectivity showed a completely contrast pattern for both species. For Quercus branti, as you can see, the most of core habitat, even though with increasing the age effect, this connectivity looks more stable. But for the Quercus macrantera, it might be in the higher risk of fragmentation, and it's supposed, as you can see from the LGM to the future, declining the ecological connectivity. Appeared, and the discontinuity of the connection and breaking the habitats to more smaller and isolated patches is appeared. The result of the zonation and overlap analysis plus the stability analysis showed that for Quercus branti, the top priority habitats is located in the central of the Zagros and south and part of the north. But the stability analysis for a refugee zone is in the northwest of the Zagros, which it looks to remain stable through the LGM. But for the Quercus macrantera, the top priority of the habitats in the central and the eastern of the Hyrcanian forest. But the climate refugia concentrated in the eastern of the Hyrcanian and western of the Alborz, which through the extending into the south of the Caucasia. The analysis of the overlap between the protection area and high suitability range showed for both species is quite limited, and majority of the priority area is outside of the protection. It's almost 70% for both species. So altogether, we can say that integrating of the long-term refugee zones and assessing the efficiency of the protected area is crucial for designing and identifying the area of the high ecological resilience and conservation strategy. If you want to have more information about this paper, I introduce you. Thank you for your attention.
Thank you very much thank you very much for your talk. And our next speaker will be Michał Dalikta, who will tell us more about estimating current and future bioclimate sustainability of alien tree species, Ailanthus altissima, on the tree-related land cover types in Europe. Here. Okay.
So good afternoon. My name is Michał Dalikta. In this hopefully short presentation, I will show you how the climate change made the special range of Ailanthus altissima, so island tree species, more suitable on tree-related land cover types in Europe, and what it could mean for selected types of ecosystem services provided on those land cover types. So, a short reminder about invasive alien species. Invasive alien species are currently considered as one of the main ecosystem threats and main cause of degradation of biodiversity. Their rapid spread and intensive reproduction beyond native range. They usually outcompete native species in disturbed ecosystems. They alter ecosystem structure, reduce this biodiversity, and decrease resilience. They also reduce key ecosystem services, so in timber, water regulation, and recreation. One of invasive alien tree species is Ailanthus altissima. It is native to southeastern Asia, introduced in Europe in 1950s as ornamental species. Currently present in most parts of Europe. It is on list of species of union concern. Its invasiveness, it is described by, for example, the seeds production. So it reproduces generatively and vegetatively. It has some allelopathic properties. It tolerates drought, poor soils, frost, and air pollutions. And their main dispersal pathways are through the cities, to transport corridors and forests. So we usually consider invasive alien species as our enemies, but I think it might be some controversy that in selected urban areas, in selected situation, we should consider them as beneficial to selected areas. So in urban areas, they provide shade and cooling effects. They support air quality. and they could establish in degraded habitats. But it has some, of course, costs. So the invasion could suppress native flora and suppress, as I mentioned, the selected ecosystem services like reduction of timber, water regulation, recreation service, infrastructure demand, and some allergenic properties. So we have short-term urban benefits versus long-term ecosystem ecological risks. So main question we asked when preparing this study is which tree-related land cover types face the greatest bioclimatic invasion risk currently and through the end of century. To do so, we have made three tasks, 3 aims. So first one was to model current and future bioclimatic suitability across Europe under multiple climate change scenarios. Second one was to analyze how bioclimatic suitability varies across tree-related core land cover classes. And last aim was to identify which land cover types face the greatest increase in invasion risk, providing primary data for ecosystem services evaluation at European scale. So our study area covers Europe. We gathered occurrence data from GBIF. At the time of the analysis, we had over 100,000 records available. But as we know from previous speeches, for example, this one about the big data, we shouldn't use raw occurrence data because it's biased in some way. So to reduce this bias, we proceed with filtering of GBIF records. So we removed incorrect coordinates, like plain zeros in coordinates. Some radius around country capitals, because in GBIF, when the location is unknown, they usually they sometimes put them in around country capitals, around country centroids. We also got some records located within oceans. We also removed records with spatial uncertainty more than 30 Earth seconds. It was connected to the model, to the environmental data I will soon explain to you. We removed also records with repeating coordinates outside the study area, and spatial correlation was also removed. So from those 134,000 records, we got almost 24,000, so 17% of our initial number. And later with those records, we processed with modeling using Maxent and climate scenarios. So to model spatial distribution modeling, we used Maxent algorithm, which performs nice with presence-only data, because in case of using occurrence data, we usually have presence-only. Some from those occurrence records, we used the 2-to-1 ratio of occurrence records versus background points. And to validate the model, we used 10-fold spatial cross-validation with blocks in size about 100 kilometers. About environmental variable selection, we have 19 bioclimatic variables from WorldClim, and we retained only 5 of them after removing those correlated with person correlation above absolute value over 0.7 and variable inflation factor less than 10. So final set was BO2, BO11, BO15, BO18, and BO19, which is mean diurnal range, mean temperature of the coolest quartet, precipitation seasonality, precipitation of the warmest quartet and of the coldest quartet. So usually when when conducting special distribution modeling, we talk about suitability, and this suitability is in range of 0 to 1. But in this time, I wanted to check something and experiment with threshold values. So the threshold value of 0.578 was then used to calculate maps with threshold deviation trends. So above the threshold, we consider the area as suitable for the species, and below, unsuitable. We also calculate uncertainty, but during this presentation, we don't have time to talk about it. So the previously mentioned projections were about current climate, but we also have some models for the future climate. So we use 3 general circulation models from CMIP6 framework, assembled by Median, in 4 periods, in 4 future periods: 2021 to 2040, 41 to 2060, 61 to 80, 81 to 100. And when talking about the future climate models, we also talk about the models of the scenarios of carbon dioxide emission. So the most optimistic, so the strong mitigation of carbon dioxide is SSP 1 to 2.6, and high emission scenarios is SSP 5 to 8.5. So about model performance. We have a couple of metrics we use to describe model performance. So area under curve, continuous Boyce index, and Ruskey statistics. And we describe those values that we have, that model performed quite okay, quite good. And this is important thing. We have a dominant prediction, BO11. So it described, it contributes almost 100%, which might sound bad, but later I will describe, in next slide I will describe what could cause this effect. So BIO11 dominance contribution might partially reflects that after reduction of those correlated variables, it represented several correlated winter temperature variables that were removed. At the beginning and in the title we have something called 3 related Kórnik cover classes. So we selected 4 of them. So CLC Kórnik cover 3.1 forests, which provide timber carbon sequestration, water regulation, biodiversity and recreation. CLC 3.2, so scrub herbaceous vegetation, which is the dispersal stepping stones of the species living in the urban areas. We have also CLC 3.3 open spaces. As we know from the literature, island open canopy facilitate colonization of islands altissima. And CLC 1.4, so artificial vegetated areas. So first results of the modeling, so the current bioclimatic suitability. And we can see, I think, clear gradient from southern and western Europe was above the threshold, and northern and eastern were unsuitable, so below the threshold. So currently 16.5% of Europe is suitable. And the mountain ranges act as persistent climatic barrier. For the future projections, in the SSP 1.1 to 2.6, so low emissions, we have moderate northward shift in suitability. Natural forest and land cover types remain below the threshold, and the boreal zone remains unsuitable and strongly negative. So here we have 21 to 40, 41 60, 61 80, and 81 to 100. And in case of SSP, so high emission, SSP 5 to 8.5, it is quite divergent from the previous one, and becomes spatially apparent from 41 to 60. And by end of the century, nearly the entire continent is above the threshold. Only northernmost areas and alpine peaks remain strongly negative values, so below the threshold. And here's fast comparison of different scenarios. So in case of high mitigation of SSP 1 to 2.6, we have at the end of the century only 28.2% of the Europe is suitable. And in case of SSP 5 to 8.5, over half of the Europe is suitable for the Islands Altissima. Now about trends. So here we have only SSP 5 to 8.5 because we have weak and non-significant trends in this case. In case of forests, we have largest total shift among three related classes. The trend approaches but does not consistently cross threshold at pan-European scale by end of the century. In case of shrubs and herbaceous vegetation, it is the most consistent improvement under SSP 5 to 8.5 among CLC 3 class subclasses. And in case of open spaces with little or no vegetation, we have most negative intercepts across all examined land cover types. Improvement rates, it's comparable to the previous one, yet high altitude and high latitude concentration cause slower absolute climate change. And open canopy structure is enabling potential colonization under suboptimal bioclimatic condition, which reduce competition relative to closed canopy forest. And about artificial and non-agricultural vegetation areas. So, as we mentioned previously, it is persistent incubated habitat. The propagules disperse into adjacent forests from this area. And to sum up this part, we have a question of whether forests are at most at risk, to answer our question at the beginning. So the already suitable areas are a Mediterranean forest, lowland temperate forest, transition zone, which would cross threshold under high emission is Atlantic forest and eastern lowland forest. Emerging risk, so future exposure is boreal forest with fastest rate of suitability increase. And we have refuges, so alpine forest, which remain below threshold across all the scenarios. It's small table to sum up things about ecosystem services at risk and risk under these scenarios. And what could we do with the bioclimatic suitability models? So we could use them as primary data for ecosystem services evaluation. So currently limitation in those evaluations, the evaluation relies on static proxies. There are no temporal dimensions included. The invasive alien species impact is largely absent from the ecosystem services frameworks. We have seen the data gap, so lack of spatial primary data on invasive alien species distribution across land cover types. And we suggest what bioclimatic suitability models can provide to ecosystem evaluation. So currently, ecosystem services rely on the static proxies. No invasion dynamics over time is included. And time dynamic suitability models integrate invasion risk directly into ecosystem services assessment. And Islands Altissima suitability could be spatially explicit indicator of biological and land degradation. Some conclusion to sum up things and this presentation. So the Bio 11, so main temperature of the coldest quarter is dominant control. Suitable area expands from 16.5 to over 58%. Artificial vegetated areas show the highest current suitability, forests show the largest projected suitability gain, and this study could provide time dynamic spatially expert primary data on invasive alien species bioclimatic suitability. Thank you.
Thank you very much for your talk. And now our next speaker will be Natalia Mazurek. The title of the talk is The Age of Managed Deciduous Forest Stands Shapes Opposing Trends in Biodiversity, Lignin Gains, Plant Loss.
Hello everyone, my name is Natalia Mazurek. I'm a PhD student at the Botanical Garden at the University of Wrocław. Today I will talk about the influence of stand-aged in managed oak-dominated forests on species richness of vascular plants, bryophytes and lichens. Forest ecosystems are dynamic and their biodiversity changes over time as stands develop. In this context, stand age is an important factor shaping species diversity. Forests are not static systems. They constantly change over time. As a forest stand ages, its structure becomes more complex. New microhabitats appear and old ones disappear. To better understand these changes, we asked a simple question. How does stand age affect different components of forest biodiversity? We focused on three groups that differ strongly in their ecology: vascular plants, bryophytes, and lichens. To answer the research question, we conducted our study in southwestern Poland in Borys-Tobrowskie mesoregion. This is a lowland area with large forest complexes and about 50% forest cover. And it is representative of Central European conditions. This makes it a good model system for studying managed forests. Importantly, these forests are not natural. Most of them are managed, and truly natural forests are very rare in this region. This means that forest management plays a key role in shaping biodiversity. Even though coniferous forests dominate in this region, we focused on deciduous forests. This is because they support particularly high biodiversity and a wide range of ecological niches. Among deciduous forests, we focus specifically on oak-dominated stands. Oaks are key species in many forest communities in Central Europe. At the same time, they are economically important and often intensively managed. This makes them a good model system to study how biodiversity responds to forest development over time. This study included 4 forest types, all within oak-dominated systems, but representing different habitat conditions. On the slide, you can see their names and number of plots assigned to each type. Their spatial distribution across the study area is shown on the map. As you can see, these forest types are not evenly distributed, but form a mosaic across the landscape. This variation is important because it creates a range of conditions in which biodiversity can respond to forest aging. To capture this variation, we studied 100 forest plots. These plots covered a wide range of stand ages from 40 to 180 years. For each plot, we recorded 3 main groups of organisms: vascular plants, bryophytes, and lichens. This allowed us to compare how different components of biodiversity change along the forest age gradient. We collected data in a structured way across different forest layers and substrates. Vascular plants were recorded separately in different forest layers, including tree, shrub, and herb layers. This allowed us to identify where most of the plant diversity is concentrated. Bryophytes and lichens, in turn, were recorded on different substrates, on tree trunks, on deadwood, and on the forest floor. Species on tree trunks were recorded on one tree of each species occurring in forest layer, whereas species on deadwood were recorded on the largest log, the largest stump, and on woody debris within each plot. By combining vertical layers, the substrate types, we were able to capture a detailed picture of the biodiversity. We then analyzed these patterns using several statistical tests and multivariative models. Now let's look at what we found. Before looking at the effect of age, it is important to understand how species are distributed in the forest. First, when we look at the total species number, vascular plants clearly dominate, accounting for about 2/3 of all species, while bryophytes and lichens contribute smaller but still important proportion. However, their distribution differs. The high species richness of vascular plants is mainly concentrated in herb layer. In contrast, bryophytes are more evenly distributed across substrates, and lichens show a different patterns. They are mainly epiphytic and very rare are occurring in the forest floor. This already suggests that different groups depend on different microhabitats and may therefore respond differently as the forest ages. When we look at the relationship between stand age and species richness, a pattern begins to emerge. In general, species richness decreases as the stand ages. This negative relation is visible for vascular plants in the shrub layer and in herb layer, and also for bryophytes and epiphytic bryophytes. At the same time, lichens show a tendency to increase with stand age, although this relationship is not statistically significant. So even at this stage, we can see that forest aging does not affect all groups in the same way. So some groups decline while others may benefit. However, stand age is only one part of the story. Forest ecosystems are influenced by many other factors, including habitat conditions and landscape context. To account this complexity, we included these variables in multivariative models. And when we included additional variables, the picture becomes more complex. The negative effect of stand age remains only for bryophytes, including epixylic ones. At the same time, a positive effect appears for epixylic lichens associated with deadwood. Interestingly, the effect of stand age on vascular plant is no longer significant. This means that what initially looked like a clear age effect is actually influenced by other environmental factors. In other words, stand age matters, but it does not act alone. Its effect depends on environmental and landscape context. To explore it further, we also looked at changes in species composition. This figure shows how species composition changes in relation to stand age and other environmental factors. Among the analyzed variables, stand age has a relatively strong effect, particularly for bryophytes and lichens. In older stand, their distribution becomes more similar, while in younger stand, they are more clearly separated. This is probably because conditions in older stands are more stable, while in younger stands they are more heterogeneous. In contrast, vascular plants are influenced more strongly by other environmental factors, such as site conditions. So different organisms respond differently, and stand age interacts with environmental conditions. So what does it mean overall? It means that stand age does not drive biodiversity in a simple or a uniform way. Instead, it interacts with environmental conditions and affects different groups differently. Conclusions? To conclude, forest aging does not lead to a simple increase or decrease in biodiversity. Instead, it reshapes the community. Some groups decline, while others benefit. In particular, some organisms associated with dead wood, such as epixylic lichens, tend to increase with stand age. At the same time, other groups, including vascular plants and many bryophytes, may decline. This shows that stand age is an important factor, but its effects depend on other environmental conditions in managed forests. In other words, forest aging changes not only how many species we have, but also which species are present. In the future, we want to better understand the role of specific microhabitats. We will take a closer look at their characteristics. For epixylic species, we will distinguish between different substrate types, such as logs and stems, and consider features such as surface area and stage of decay. For epiphytic, we will also consider the properties of host tree, such as species, diameter at breast height, and other relevant characteristics. This will help us to better understand the mechanisms that shape biodiversity in forest ecosystems. To summarize, the key message is simple. Biodiversity responses to forest aging are complex and group-specific. Stand age is only one of several important drivers. Thank you for your attention.
So thank you very much for your talk, and our last but not the least speaker will be Sebastian Bury with the presentation entitled Contrasting Spatial and Temporal Effects on Climate on Reproductive Threats of Scots Pine.
Hello everyone, my name is Sebastian Bury and I'm working in the Forest Biology Center. And since January, I'm studying the impact of climate changes on reproductive of trees. And today I will present the part of my studies about the contrasting spatial and temporal effect on reproduction of trees. So, according to the Copernicus data, we see the sharp and the strong increase of global temperature. And last year was the third of the warmest year in this record. And in the last years, we even reached more than 1.5 degrees Celsius degrees, more than pre-industrial average. And the same negative trends we observe for precipitation. And in many places, including Poland, they predicted that big decline in the rainfall by the end of the century. And climate changes can impact human well-being, also economy. But what is important for us, also the natural ecosystems, including forests. For example, by the decrease in biodiversity and with the shape of the natural disturbances like insect outbreaks and fires. And not all ecological processes react on the climate changes in the same speed. For example, phenology like flowering happens fast, while the very important structure like soil impact, we can observe these changes due to the centuries. And it creates a very dangerous lag because some of these processes can happen lower and not so fast like climate changes. And some authors use only spatial effect in the studying of the impact of climate changes on some processes. And spatial effect is the effect when we study like in here, like we have a USA and we have a part of USA with very warm and very wet climate. And here we can see that trees grow very fast, much faster than in the cold and dry climate. This is blue color. And here is three scenarios. In the first scenarios, the spatial effect, which based on the long-term means of temperature of or the precipitation, what is connected with the adaptation of trees to the local environment, act in a different way that temporal effect, which include also the changes in the weather, like we can have a hot summer, like cold summer next year. And in the second and first scenarios, we have the situation when it acts differently. For example, in the second scenario, temporal effects act like spatial, like we have an increase in both of them. And in the third scenario, we have a still increase in the spatial effect, but decrease in the temporal effect. And temporal effect is connected not with adaptation, but with plasticity to the changes in the weather. And this same author collected the wood core by the Pressler borer. And here we have an individual population growth under the year. And they connected with the climatic data. And they compare the spatial effect with temporal effect. And as you can see, the spatial effect, this blue line, is totally different from the temporal effect based on these wood samples. So we know how tree growth can react on the climate changes, both this temporal and spatial effect. And it's a big lack in the knowledge about how temporal and spatial effect impact the cones and seed traits. And today I will focus on the two traits, on the cone and seed mass. And these two traits are important. For example, heavier cones in general have more space for the seeds, so there can be bigger seeds and more of them, and also better protect from the some of the negative effect of the ecosystem. And heavier seeds in general have more nutrients for embryo and are more resistant against drought. So the aim of my study was to study a spatial and temporal effect on the seed and cone traits, and I hypothesized that both of these signals will be important, but will be act in different way. Additionally, I also choose which patterns can we observe for different climatic variables, different reproductive traits, and different season important for the species. I use Scots pine as a model species. It's a species with very big range and is very important for many countries, like wood productive trees, including Poland. And Scots pine and Poland are a very nice area because Poland has very big genetic diversity. And it happened after the glacier era, and many haplotypes from Europe mixed in Poland. And I think that it is very important for my studies because we should include this diversity in the interpreting of my results. But if you want to know more about these studies, you can talk with wonderful Martyna. And I analyzed the data from 184 localization across Poland in Scots pine forests. And I use two data, mass of one cone and mass of 1,000 seeds. And this data was collected by Forest Research Institute from 2010 to 2024. And I use two climatic variables, mean temperature and climatic water balance. And for spatial effect, I use long-term means from 1950 to 2009. And for temporal effect, I use weather anomalies for this same period, like biological data was obtained. And I make the analysis for seasons important for Scots pine phenology. The time is important in my studies, so the year T is a year when seed dispersal happens, but all cycle started 2 years before in year T2, and it's a year of flower bud indices. In year T1 we have a very important pollination, and in Poland it happens in May and June. And year after the pollination, fertilization happens, and it's year T. And in my studies, I use different seasons, but I will be presenting today the results for the springs, so for the pollination and fertilization. And here I have a map for temperature for the climatic water balance in Poland, and we see that central Poland has a very high temperature and very low climatic balance. In the area close to the Baltic Sea and in the mountains we have cold climate, and the climatic balance were higher. Let's move to the results. In y-axis we have a mass of one cone in both signals, and for spatial effect here I have a long-term means for temperature, and here I have spring temperature anomalies for all these years. But it's a mean. And we see that in the colder climate, the cones are heavier than in the dry one. And in the year when we have a pollination, it's very like cones prefer the cold weather. Let's move to the climatic water balance. And here I have a significant level only for the spatial effect. And we observe that in wetter climate, the cones are heavier than in the dry one. Let's move to the mass of seeds. Here I also have only important significant results for spatial effect. And seeds prefer the warm climate. So in the warmer climate, we have heavier seeds. And the same like cones, seeds prefer wet climates. And I also have a significant effect for the temporal effect. And droughts during the fertilization limit the growth of the seeds. So in the drought springs, we have smaller seeds. So I summarize that briefly. So both seeds and both cones like the wet climate. However, seeds prefer warm springs, and cones prefer cold springs. And for temporal effect, the high climatic water balance during the spring, when fertilization happens, promote the mass of seeds, while low temperature in spring, in previous springs during pollination, promote the heavier cones. So I confirm that spatial effect and temporal effect are both important for the seed yield prediction. However, the results are different, and also the results differ between the seasons, between the different traits and climatic indexes. So my results are context dependent. And in general, the consistency between temporal and spatial effect tell us that temperature was a main driver for the cone mass structure, for the mass of the cones. It's a year before, during the pollination. And it's a foundation for the heavier seeds. But for seed mass and seed fill, more important was the climatic water balance because of this consistency between spatial and temporal effect. So I have two conclusions to management practice. So the forest manager should include spatial data with the temporal monitoring to better predict the seed yield and also the natural regeneration of the forests and the future species composition. And the second conclusion was that origin matters. As we saw, the seeds are very sensitive for the droughts in the springs during the fertilization. So maybe foresters should choose the best origin of the seeds and plant it in the and sow it in the particular localization. But it needs to we need to cooperation between ecologists and the genetics to choose the better origins. And my take-home message is that effect of climate on reproductive traits depend on signals, so temporal or spatial, but also depends on season and particular traits, and also climatic indicators. At the end, special thanks for all co-authors and funders from Forest Biology Centers, from Forest Research Institute, and from Adam Mickiewicz University. And thank you for your attention.
Okay, thank you very much for your talk. Sebastian Bury was our last speaker, so now it's time for discussion. So I encourage you to ask a question to the presenters. Okay, please.
A question for the guy working with the Ailanthus.
Okay.
I don't remember the name, sorry. No, first of all, very nice presentation, very nice work. I'm wondering, you at the end told that the next step should be the suitability mapping of Ailanthus, for example, around Europe. So I'm thinking, what could be, apart from what you have already studied, other factors that you can include in the model?
So usually in spatial distribution modeling, when we are modeling habitat suitability, because I was modeling bioclimatic habitat suitability, we use data about soils, about elevation, about digital elevation models. Be the next step, but in my case, when I'm modeling future data, we cannot directly model future. We can model future soils, but not as accurately as I would say about the climate. Climate is also connected with the soils, and so in this way we can model future soils, future elevation models, and etc.
I understand. I was not referring to future
okay.
Because obviously it's difficult to model a future soil or whatever. So okay, I be one of the most important parameters for Ailanthus, which is a species which is looking for degraded soils and environment. Maybe just as a suggestion, probably what will be important for the suitability for such a species can be the distance from roads, from line ferry, because it's from my experience, the place where the invasion of Ailanthus started. So in the case that in the future you would like to do this kind of study, maybe this can be a parameter.
So small sneak peek, I'm working on and using LiDAR data also, density of the crowns and distance from the different factors. Thanks.
Okay, thank you. Maybe some other question for others presenters? Okay.
I also have a question for Michał, because I was also working with data from GBIF, and you mentioned that some records were marked as present in ocean or something like that. That is, I also stumbled on this, and those records are usually some metagenomic projects. And I would check if this data is reliable, because I had some issues with species level identification with metagenomic samples, and I would look in the primary studies that publish this data, because they can be somewhat misleading because of that. But of course, that was those measures that you took, you probably remove all the biases, but I think that's just something to have in mind.
Okay, thank you.
Okay. Any other question for our presenters?
Depending on the age of the forest stands. You said that when we are talking about the younger stands, the site conditions are more important than the age, if I understood it correctly. If so, I wanted to ask what are the site conditions that are the most important? Because is it like the fertility of the soil? Is it the water regime of the stand, or like? And why do you think that when we go to the older stands, that the age matters more? Is it because of the microclimatic conditions that are created by the older stand, or why does it change as the stand ages?
Okay, thank you for your question. It's not exactly like you say. So for vascular plants, the other variables are more important than age. And this is soil fertility, soil moisture, also the landscape context. I mean, if they are coniferous stands in the neighborhood of this forest plot where we have collected data, so the other variables are more important for vascular plants. And so it's not so like that in younger stands the conditions are more important than in older, just that for vascular plants other conditions are more important.
Okay, thank you very much. Maybe some other question for other presenters? Okay.
For the guy who worked with Scott Pine.
Okay.
Congratulations for your presentation. My question is, you mentioned that you used the cone mass as the seed trait, and we had the discussion about the seed quality. Do you think your measure with the cone mass covers too, like the cone quality that could be the seed quality that could be affected by the climate and the spatial traits?
Yeah, maybe it's important to talk about the seed quality, but I know the studies like, for example, heavier seeds better, like it's more like stress tolerant, like they are heavier, bigger, so they are not dry out so easily like the smaller one. But you mean like the possible to germination? So yeah, it can be connected. Yeah. So heavier seeds and better germination. But I know also the opposite results. So maybe it depends on the species. Yeah. Thank you.
Thank you. Any other questions? Okay. If not, thank you very much for your questions, because I know that it's not easy sometimes just to, but maybe, not maybe, you will have time, more time just to talk among yourself and ask a question. So I encourage you just to talk and discuss. So thank you very much, and now we can take a break.